Hello, everyone. It's Michael Shermer, and it's time for another episode of The Michael Shermer Show. This one brought to you by Wondrium, the series of college-level audio and video courses and documentaries produced and distributed by the Teaching Company. You remember them, the great courses, which are great. Wondrium brings you engaging educational content through short-form videos, long-form courses, my favorite, tutorials, how-to lessons, travelogues, documentaries. I've seen a few of those too, also excellent. And many more, covering every topic you've ever wondered about and many you've never thought you'd wondered about. Here's my example for today. By the way, if you subscribe to Wondrium through the show, which is what I'm asking you to do here, to support the show, you get 2 years for the price of one. That is, you get a 2-year subscription, which gives you access to all their content online, streamed straight through your phone or your iPad or your computer, and you can watch or listen. I always just listen to the courses while I'm out doing my thing, and you can too. It's a great source to do that. Anyway, if you go to wondrium.com/Shermer, you get 2 years for the price of one. Wondrium, WONDRIUM.com/Shermer, 2 years for the price of one. Here is my example of a course I've just started taking. I've listened to 4.5 lectures. This one is on ancient writing and the history of the alphabet. Did you know there's a history of the alphabet? I did not know. The reason I signed up for this one is because it's being taught by my longtime good friend and colleague, John McWhorter, the great linguist. And so I listened to the first 4.5 lectures, The Nature of Writing, Cuneiform, the world's first writing, how Egyptian hieroglyphics work. I had no idea how these things work. It was really interesting. The invention of the alphabet goes east. And then continuing, these are the ones we listen to the rest of this week. Lost at C, the letter C. The history of H. I had no idea H had a history. So interesting. The ramblings of R, the letter R. The unfolding of U, V, W, and F. F after U, V, W? Okay. Meditations on M, D, X, and T. And then finally, there's the last 16th lecture of the 16-lecture series. How did punctuation develop? Yeah, that's a good question. Where'd that exclamation point come from? Semicolon, colon, what? Okay. Anyway, check it out. Go to wondrium.com/Shermer. 2 years for the price of one. Thanks for supporting the show, and thanks to Wondrium and the Teaching Company for supporting my work. All right, here's the episode. Here's the magazine Skeptic I want you to check out. Go to skeptic.com/magazine. Order your subscription today, or you can go to your local Barnes & Noble or other fine bookstores that carry. We're still in print. Check that out. There's still an actual physical magazine. Some of us are still around. Of course, you can get it on digital as well. If you support the podcast, you can do that through the Skeptic Society. We're a 501c3 nonprofit. Go to skeptic.com/donate. All right, I got a really important episode for you today on the future of science and medicine. My guests are Leroy Hood, MD, PhD, who developed the DNA synthesizing technology that made possible the Human Genome Project, and is co-founder of the Institute for Systems Biology, a pioneer in the field of systems biology, proteomics, and P4 medicine. They'll describe all that later. He's won the Kyoto Prize, the Lasker Prize, the Heinz Award, and the National Medal of Science. Okay, that's some definite creds there. Nathan Price, PhD, is Chief Science Officer of the Thorne Health Tech and Professor at the Institute for Systems Biology. Selected as an emerging leader in health and medicine by the National Academy of Medicine, he received the Grace A. Goldsmith Award for his work on scientific wellness and has co-authored over 200 peer-reviewed scientific publications. Here's the new book. It's called The Age of Scientific Wellness: Why the Future of Medicine is Personalized, Predictive, Data-Rich, and in Your Hands. Check it out. It's a great read. It's from Harvard University Press. And gentlemen, thanks for coming on the show. You know why I want to have you on the show? Because I'm 68 years old and I'm getting worried. And I got a 7-year-old now. I have a 31-year-old daughter. Now I have a 7-year-old son. I got to make it into my 90s because the little guy needs a dad. And then my daughter's her children will need a grandfather. So I'm relieved. I was really uplifted, actually, by reading your book. Like, okay, there is hope to get there. Not the crazy, like immortality attempts through cryonics or anything like that, but actual things we could do. So let's start with just giving us a little bit of background. Leroy, why don't you start? You start in the first chapter, giving a little bit of background. super interesting background how you got into all this.
Well, I got into it basically when, as a young assistant professor at Caltech, I started thinking about where I wanted my scientific career to go in the future. And I had a real interest in human biology and disease. I was dismayed at that time, 1970, by its complexity, by the lack of tools we had for dealing with that complexity. And in thinking about it, I decided to develop instruments that could decipher that complexity by generating big data, lots of information on individuals that ultimately, when analyzed, could lead to insights into wellness and prevention and the things that we'll talk about later on. I went through a whole series of paradigm changes early in my career. I developed 6 instruments that basically allowed one to read and write DNA, and they served as the foundation of modern biology. And certainly they give us the ability to get big data from individual humans. I was invited to the first meeting ever on the Genome Project, where 12 of us passed opinion and came to the conclusion, 1, it was feasible, 2, we were split 6-6 on whether it was a good idea. Those opposed it because of big science and their fear it would swallow up all of small biology, which, of course, it never did. I then went on to create the first cross-disciplinary department at the University of Washington with the help of Bill Gates. And then in 2000 started the Institute for Systems Biology with a whole new holistic, global, dynamical view of biology and how you should proceed to study complexity, and applied it to wellness and disease. And then I think the big event for this conference is that Nathan and I in 2014 actually created the first precision health medicine population, 108 of our friends with big data, which we generated from the blood, from the gut microbiome, from digital health measurements and the like, to give deep insights into wellness and into disease. And this led to a company started in 2015, Airvale, that generated 5,000 customers over a 4-year period with genome data, the sequence of your 22 pairs of chromosomes and everything, and with phenome data, that is data that could let us explain how different stages of your life appeared so different from childhood all the way up to final maturity, adulthood, old age, and the like. And the phenome, as it was called, were these blood measurements, and they were all the measurements except for the genome itself. And then finally, I've proposed, and we'll talk about it later, that we could move from 5,000 in Airvale to 1 million people to really validate all the fascinating things we discovered with Airedale in a second genome project where we would analyze the people for a 10-year period doing genome-phenome analysis. And that would lead to 2 important results. One, an unequivocal proof of enormous increase in the quality of healthcare. And 2, a demonstration that one could imagine that the science of wellness and the science of prevention would lead to cost savings of the order of trillions of dollars a year in an annual budget today that's about $4 trillion.
Nice.
So that's a quick summary of my background.
You mentioned in the book you read Kuhn's Structure of Scientific Revolution about paradigm shifts. And then you also mentioned that you were a professor at Caltech. Well, that's like the Pope job. Doesn't get any better than that. And then you leave for the University of Washington. You get some Bill Gates money, start this new foundation, and your colleague or dean or whoever at Caltech says, "Have you lost your mind? You could be at Caltech the rest of your life." In a sociology of science sense, is it your sense that if you really want to launch a revolution, you got to break out of the pure academic paradigm and go somewhere, a pathway like what you took?
Precisely. I think any brand new idea almost never can be achieved in the context of an existing bureaucracy because bureaucracies are honed by the past, frankly, can just barely deal with the present and have difficulty dealing with the future. And, Caltech was a great place, wonderful students, wonderful faculty, a dream place. You'd ask, why would you ever leave? And and the two incentives I had to think seriously about leaving and finally to leave were one, the two things I cared about most, my senior colleagues in the biology department didn't approve of. One was an enormous interest in technology and engineering and things like that. They recommended I move to the engineering department, which would have been a disaster for me because I wanted biologists, not engineers, who were going to decipher this complexity. And of course, the second was my growing interest at that time, and this was the 80s and so forth, in the Human Genome Project. And they initially, like 80% of the biologists in the US, were absolutely opposed to it. And, I had to ask myself if I really wanted to push this revolution I saw coming, I'd have to do it in a place that embraced the roots that catalyzed this change in wellness and prevention that was absolutely mandatory for improving healthcare in the future.
Interesting. Yeah. It's a similar story. My friend Christoph Koch, who is a neuroscientist at Caltech, he had the Pope job too. And he left and went to the, Allen Brain Institute, the Allen with the co-founder of Microsoft and so on. Sometimes maybe it takes private money and just a whole new start to do something like that.
I think that's right. I actually hired Christoph Koch at Caltech.
Oh,
So
you did?
he
Oh
was a
gosh.
wonderful,
Small world.
brilliant,
Yeah.
out of the box kind of thinker. I mean, anyone who would take on the challenge of consciousness has to be incredibly brave and out of the box.
Yeah, for sure. All right, Nathan, what's your story?
Yeah, so I come into this a lot later than Lee, being about 40 years younger. And so for me, I became very interested in science as a kid, mostly by reading. So I grew up in Utah. I probably never even met a scientist until I was in college. So you might wonder how I got into this whole business. But I loved the Richard Feynman's books. I loved Stephen Hawking's books. I read all that stuff voraciously. I was very lucky in the sense that, I mean, it's going to sound awful to say it was lucky, but my grandmother's house burned down. And so when her house burned down on kind of a quirky problem in the mail, she ended up having a weekend without insurance. Her house burns down. She can't replace it. So she moves in with us. And my grandma was a really gifted uh, private elementary school educator. And so she ended up turning it into a school out of the basement of our house. So I pretty much had an incredible education from her from a young age and became, at least if you believe in all the tests or whatever, became really good at math and science and thinking and reasoning. And I owe that pretty much all to her and kind of being trained from that from an early age. So I went through school, and then I ended up studying bioengineering at the University of California at San Diego, which had a real top department, and was working on building computer models for how you process food into yourself, all the different chemical reactions, and building large scale computer models to deal with that. A lot of this was made possible from Lee. And I think one of the interesting links between Lee and I is that, Lee's the creator of a lot of these systems biology, interdisciplinary thinking, and all those kind of things. I'm very much a child of that world. So I grew up in a world where I was interdisciplinary from the beginning, where I was thinking about systems biology and the fusion of computing and engineering and biology from the very start, much because of the world that Lee had built. So So I went through the department at UCSD and was fairly prolific there. I set the unofficial all-time record for papers published out of the department and things like that, which was kind of fun. Um,
Really?
well, it was the most that we had. It was 21 peer-reviewed science publications out of my thesis, which was at least the best at the time. So actually, anyway, I won't get into that. But so I was interested, so I accepted a faculty job straight out of grad school at the University of Illinois in Urbana-Champaign. I had a top 10 rated chemical engineering department and an institute for genomic biology. But I deferred it for 2 years to do a postdoc, and I wanted to identify the person who I thought was the leader in the world that I wanted to connect with on the topic. That was Lee. And so I reached out to Lee, or actually I had my advisor reach out to Lee because I was afraid that I would be lost in all of his email. And I still remember the entirety of his response back. Not a feat of memory, but the entire response back to my application was, price looks great. That was the entire email. That's all it said. And then Lee and I connected, and I ended up deferring the position in Illinois for 2 years to go work with Lee as a postdoc, which was wonderful. And Lee and I worked on a number of interesting projects. I then went back to the University of Illinois, and Lee and I stayed close throughout that time. And we'd talk on a weekly basis, and we'd go out to his place in Montana every summer for a week, and got these beautiful ranch houses for a retreat out there. And about after 4 years, Lee reached out and was with the offer to come back. Lee had just secured a kind of a heroic level grant from the country of Luxembourg for $100 million, which helped make it possible. And in my 4 years at Illinois, I'd built out a group of about 25 grad students and postdocs, and so almost all of them moved with me back to Seattle. I became the associate director at ISB a few years later, and then we got into this area of scientific wellness that Lee talked about. About 5 or 6 years ago, Lee and I combined our lab groups together. Lee had become the chief science officer for the Providence healthcare system, and so came to me with this idea of taking what we were doing, and me being primarily a computational biologist and Lee being primarily on experimental biology and technology. We thought fusing those things together into a joint enterprise would be really powerful, so we did that. And then about 2 years ago, speaking of sometimes going into different avenues and so forth, I was approached by Paul Jacobson, who's the CEO of Thorne, where I am now, who basically had become very interested in all the science that we had done on scientific wellness, and called me up and said, if you really want to make all these things that you've discovered like operational in the world, you need to tie them all together into one thing and make it economically viable and scalable so you can take it to millions of people. And so I ended up moving about 2 years ago to become the CEO of a startup AI startup company called Longevity, but then merging that with Thorne Research to form this new company, Thorne Health Tech, which we IPO'd in 2021, which is a company dedicated to the delivery of scientific wellness out to the world. And we now have about 5 million customers so far and work with 46,000 healthcare providers.
Amazing. I love stories like these that are not non-traditional careers, as I guess they call them. Okay, what's the problem to be solved here? What is systems biology? What's wrong with reductionism? Reductionism has been so successful in science. What's the problem here?
I think the problem is this complexity. Reductionism is the approach where you take an element of a complex system and you study that element in enormous detail. And this is classically what small science did up until systems biology came along. And the deficiency in that approach is studying one element in a complex system gives you no insight into how the complex system starts to a first approximation. And of course, what systems biology says is, look, what underlies all of the complex responses and phenotype of human beings are biological networks. And what we have to do is first identify the components in these networks and then study their dynamics if we're to understand how the brain works or how muscles work or these kinds of things. So systems biology is all about taking a global, holistic view of a problem you want to study and not thinking just in terms of one or two elements, but thinking in terms of the networks that encode the information that is responsible for that particular phenotype and so forth. And this idea of systems biology then has really pervaded the world. I would guess, realistically, there are probably 200 institutions across the world that have the name systems biology, institute, department, center, whatever. And it is a new way of thinking that really now pervades biology. But I think the most fascinating part of it really is it should be applied to any kind of complex problem that you have, whether it's psychological or sociological, physiological, or what. I mean, systems approaches are exactly, frankly, what politicians should learn about so they could think about something more than tomorrow. There is a future in 10 years and 50 years, and what are we leaving our kids? That is what's missing today in politics for the larger part. So it's just a global, holistic approach that I think is compelling in the results it's produced in terms of gaining understanding of complexity.
So to take something like DNA, it's good that we had Crick and Watson and drilled down to the molecular structure. But that's reductionism. That's very successful. But that's just the start. There you have to then build back out to the phenotype and the entire body and how it interacts, systems within the body and other systems socially and so on and so forth. That's what you mean by systems.
Yeah, the DNA, the first thing you had to figure out is what are the elements of information that DNA encodes? These were called genes. And once you'd identified the 20,000 or so genes, you had to figure out how do these genes connect together to make these networks? And then finally, you had to watch the networks operate during the dynamics of what you were studying, thinking in the brain, what have you, to understand how they actually executed their function. And the other thing about systems that's really important is they operate across multiple scales. So the system can be thought of at the level of one molecule, at the level of one cell, at the level of an organ, or at the level of the whole organism. And how do you connect together these different hierarchical levels that operate at different times in space and time and everything? That's the essence of systems biology.
Yeah, Nathan?
Yeah, and I think you can approach that problem of complexity. It's so interesting because if you're thinking about reductionism, the challenge that is before you is daunting, right? You're trying to find something that doesn't exist, but conceptually, it's a very doable thing. Like you can get your mind wrapped around the notion that A, T, C, G, that's a letter of DNA, and it just extends 3 billion letters, right? You can't think about what the whole 3 billion is, but discovering the structure is something that is, physical, tangible, and easy to conceptualize. The flip side of systems biology, when you're trying to put together all the pieces, you really start to get into areas that are beyond our comprehension pretty quickly. We talked about Christopher Koch earlier, right? Consciousness, right, which is a really high level of an emergent systems phenotype that we have, I would argue, very little clue about, how to talk about that at this point. And there's all these intermediate layers. So there are areas where we can get into, networks and systems predictability. And there's been all this tension between molecular biologists and systems biologists, especially in the early days, because molecular biology sometimes can feel very satisfying and concrete, like here's the protein, here is its sequence. And when you're building a system, you often have a very complex relationships amongst all these, and you get a predictive accuracy, and you're often trying to just up that predictive accuracy. So you go from 60% to 80 to 85 to 90, and you're just trying to capture more and more of it. And so I think that's one of the big differences is just the level of how easily it is for us to get our minds wrapped around. But with the new tools with AI and all the large measurements, systems biology is where the action really is in many ways now, because that's what it's all about is how do you take these tools and then control the phenotypes that we care about, end disease, reverse it, extend your health span, slow aging, all these things that we want to be able to gain power around.
Let me pick a quotidian example of weight loss and diets. Why are diets so faddish? They go in and out. And then even during a particular fad, like this is the diet. And then, a million people try it and it works on, 10% of them or 25%. Why is there so much variation? Is this because even if you're controlling for genes, there's just so much more at work different between people, even on the same diet, same exercise, and so on? They just have different responses.
Yeah,
So I think the answer, go ahead, Nathan, if you've got.
yeah, I can give you a couple of specific examples of that. So one is that in the Airvale population, we actually looked at people who went on to lose weight from those who didn't. And we asked the question at baseline, if you look at all these thousands of measurements that we've made, was there anything that was predictive about whether or not people would go on to lose weight? Then we looked at metabolites and proteins, and once you normalized for BMI, we couldn't find any predictive power. But we did find predictive power in the microbiome. And it turned out there were two features that were important. So the first was how fast was your microbiome growing? And there's a trick you can do on sequencing where you pile it up around the replication fork as you go around. And the more stacks that are at the beginning compared to the end, this is called peak to trough ratio, the faster they're growing. So it's a cool little trick you can do to estimate a growth rate from a static measurement. So if you do that,
Nathan, hang on. I'm going to pull my Back to the Future. Doc, English, please. What did you just say?
Yeah, sorry, I did. We don't need to get into the details. There's a trick you can do where you can estimate the growth rate. So if your bacteria, so if they're growing faster, so every calorie that you eat is a calorie either for yourself or your microbiome. If your microbiome's consuming more, easier to lose weight. The second big factor that made a difference is if the genes in your microbiome are more likely to break down complex carbohydrates, right? So you eat a sweet potato or something, and you break that. So some microbiomes will break that down into simple sugars that will spike your insulin more. It's harder to lose weight. But if you have a microbiome that will break down those complex carbohydrates into short chain fatty acids, then it's easier to lose weight. And so that was quite predictive. The correlation on that would be 0.3. So it captured a fair amount of variance in terms of that. So that's one. The second big example, which isn't explicitly weight loss, but just to give you a sense for what happens. So if you were trying to lower something like LDL cholesterol, so it turns out you can predict from the genome a level of LDL cholesterol in the blood, not knowing anything about diet or lifestyle or anything about the person. Some of that is just genetically encoded. So one of the things we looked at is if people were going through a wellness program, could they lower their LDL cholesterol without a drug? And some people could and some people couldn't. So it's exactly what you said. Well, people do this and it's kind of a mishmash, and some people do and some people don't, and there's a weak effect. But it turned out that if you looked at their genetics, you could predict who would lose and who wouldn't. And so what you had was a strong effect if there was a gap. So if you had high LDL, but your genome predicted low, you could move it. But if you were high, and if your genome predicted you high and you were high, then you couldn't. And so that gap, just to give you an idea, is an incredibly useful tool to know what you can change easily from what was going to be hard for you to change. And it introduces an entirely new category of variable into medicine, because we don't use that at all today, because doctors by and large don't use the genome. And so what that means then is you can introduce a totally new category where you look at your genetic potential versus your actual, and it can give you a roadmap to what will make the biggest difference for you in terms of making a lifestyle changes that will affect your health. So that's a big deal.
Unbelievable. That's super interesting. So, whether you should go on a statin or not, No, the doctors just say just everybody should take a statin, just if if you're even remotely close to the top of where you should be. But that's just so crude. You got to do but you can't just do a 23andMe to find that out, right? It's got to be something much more in depth.
Yeah, you have to have both the blood measures and the genome to be able to do that. So we are
So
working on building that into a
yeah. So
product.
what would it take to get that to be a common thing? Go to the doctor, I get my genome checked.
It would take a program like Aerovale where you could have many of these features looked at, including many of the blood analytes that make the correlations Nathan talked about. So program today doesn't exist, but a few places are starting to begin to create programs that will evolve into programs where you can get that kind of information.
But I mean, if you guys have your prophecies come true in this book, hopefully soon, that kind of test should be available to everybody in every doctor office. And so the doc says, well, your cholesterol is high, your LDL is a little high, but before we give you the statin, let's run the test and see if it's going to work or not.
Yeah,
that should just be commonplace.
So it should be commonplace, and that's exactly the reason I've proposed this second genome initiative, where we take 1 million people and for 10 years we do all these analyses. So we'll have all the correlations for 150 different genetic risks, and we'll have all the correlations with the phenotype kind of information. And we can show unequivocally how this transforms the quality of your life. And I'll guarantee you we're giving drugs to many people who should never be getting drugs. They would be in a place to manage their excesses themselves just by diet and exercise.
You had this line in the book, the 10 most popular drugs in the U.S. work for only about 10% of treated people. What?
Isn't that an absolutely striking statistic? I will say that people argue about maybe the number should be higher, but even if it's 20% or 50%, it's still appalling because those that it doesn't work for are paying for the drug, often expensive, or their healthcare system is, and B, that you're exposed to all the side effects of the drug and everything. So a critical thing that'll come from the Million Person Project within the first few years is I think we'll have blood biomarkers that can tell us unequivocally for many of the drugs which people are going to be able to respond and which not. That is something pharma companies would hate because they like this idea of one drug for everybody. And of course, that's becoming increasingly impossible. But the idea that only 10% of the people could safely use the drug, I think, is an anathema to many. But it is, in fact, true for some of the drugs that are out there.
Yeah. Leroy, could you, one second, Nathan, Leroy, can you turn off your email because we're getting that loud ping every time you get a new email. Go ahead, Nathan.
Oh, okay.
Yeah, and another factor I was just going to mention that has come out very recently is that the first estimate is that your microbiome will eat about 13% of the drugs you take. So it transforms them. So the initial estimate was really fast, and it's a different percentage. It's different drugs for different people. It's not just one class. And so what that means is that you could be taking a drug, and if you have the wrong microbiome, it could be changing that compound, and you're not even on the drug that you think you're on. So this is a big problem for drug companies to start thinking more systemically.
Can you explain what the microbiome is and what I could do about mine, or listeners can do about theirs?
Absolutely. So the microbiome is one of the hottest areas in health now and has just emerged quite recently in terms of our understanding. So the microbiome are all these small species. So these are bacteria and other small organisms, and they live on your skin, they live in your gut. They're basically an envelope between you and the outside world. And so everything that you take into your body, whether that be food, a supplement, a drug, it comes into your body, it passes through the microbiome before it gets to you. And if you go back in time, certainly when I was a kid, and if we think back earlier, medicine was always kill germs, right? You got to kill the germs, and we took antibiotics like crazy, and that causes all kinds of problems. One, it creates antibiotic-resistant bacteria, which is the whole thing. But the other thing that it does is it decimates your microbiome, and it turns out that your microbiome is critical to many aspects of health. And so you can get access to a lot of information about your microbiome now. So for example, Thorn Health Tech, where I'm at, so we have a test on the microbiome. What are the kind of things that you can learn off of this? So you can find out from your microbiome, one, how it affects your digestion. So you might have microbes that are making too much ammonia, for example. That will cause your stomach to be too basic. It won't be acidic enough to really break down your food the way you want. You can evaluate that. You can look at how the microbiome affects things like probiotics that you're taking. So if you're taking a probiotic, you want to know if it's actually engrafting. You can see that. You can identify gut dysbiosis. So if you're having a problem, we did a trial on people with irritable bowel syndrome and showed that over the period of a month, you can get a test, do personalized interventions, and you can resolve the symptoms of irritable bowel syndrome in most cases over the course of about a month, and so forth. So you can dive into this. The second thing that I'll just mention is we've also really worked hard to try to make it easier for people to do a microbiome. So the traditional approaches they have a step in there that people usually don't like, which is, I don't know if you've done your own microbiome, you know
No,
where I'm
I
going
haven't,
with this.
but I think I know where you're going, but I haven't done this.
You know where I'm going. So typically what you have to do is you've got to poop in a bucket or on a piece of paper or something that comes in a kit. Got to take a little shovel, you got to get a little stool sample, put it into a vial, close it. Some of the tests require freezing, and I don't know what you keep in your freezer, but probably not medical samples. It's probably might be something you eat, I would imagine. So there's barriers to people doing this. So we got together and we sort of just thought, well, what's the easiest way we could possibly get a fecal sample? And so we invented something called the microbiome wipe, which is exactly what it sounds like. It's basically toilet paper made out of a special polymer, and you can wipes. You just do like what you do on a daily basis, but you can drop that into a vial, close it, and if you shake it, it will dissolve away in 10 seconds. And then you release a salt solution that will preserve the DNA. And we showed that worked. We published that in Frontiers in Immunology, peer-reviewed literature last year. And so what that does then is that it allows you to get a sample in a much easier way. And there was an anecdote that I loved on a different podcast with Sarah Gottfried, who's a friend and a leading functional medicine doctor, 4-time #1 New York Times bestseller, all that kind of good stuff. But she was sharing, she's working with an NBA team, and she was trying to convince them of how important the microbiome was for their health, but they would never do the steps that I just walked you through. But she switched them to the wipe, and now they've all done it. And so, there's going to be information coming out. But anyway, I just wanted to point that out because we think it's useful. And as scientists, we tend to think, all right, here's the data, right? We want the data. We say, here's the health result. We're done. And the honest truth is that you're not done at that point because you've got to turn it into something that's easy for people. And then you have to lower the barriers for people to actually adopt. Otherwise, it doesn't, in fact, make a big difference in the health of the populations. We're very focused on all those different aspects.
Okay, but let's say fast forward, it's like a pregnancy kit you can pick up at CVS, take it home, you do the wipe, and then what? So is there a program that'll then tell me I need to do this? I need to eat yogurt with the microbiome formula in it or whatever.
Yes. Yeah. So people can get this today. It's a commercial product. It's out there. And
what's it called?
So it's Thorn Gut Health Test. So it's available at thorn.com, or you can get it on Amazon as well. And so if people do this, right, you get the collections from the wipe, it sends in, then a report comes back, and then it will tell people some of the kind of things like we just walked through. So how prominent is ammonia production in your gut? How much is your gut microbiome contributing to inflammation in your body, which leads to a lot of chronic conditions? A lot of your neurotransmitters, serotonin, for example, 90+% of your serotonin that you use in your brain is actually produced in your microbiome in your gut. and you can see if you are doing that properly. A bunch of vitamins that you use are actually made in the gut, and you can see if that is happening at the level that you would want it to do. You can see if you are, if the probiotics, if you have good species of bacteria maybe that you're taking, you can see how many of them you have, and especially if you're doing something, if you're on a special yogurt, you can see if it's in fact working in engrafting. You can look for pathogens that are in there, so disease-causing bacteria, those will show up, and so forth. So there's a huge amount of information on the gut-brain axis, digestion, gut dysbiosis, all these kind of things that you can pull out of the microbiome test, and that's available to anyone today.
Amazing. Well, that's where we got to go. This is what you mean by individualized medicine. So to rewind back, I don't know, a century or so, I mean, the technology and the knowledge simply wasn't there. So they had to do these kind of large scale, okay, here's our drug, here's the problem, we're just going to give everybody that and hope we get maybe fix half or something like that. That was just the best they had to work with.
That's basically right. And today, with the existence of maybe a million different, maybe 15 different million-person genome projects, what's going to happen is the genome itself is going to reveal a whole series of diseases for which we're going to have to make drugs and so forth. For example, there are roughly 7,000 rare diseases. So these are single gene defects that have been described. And for each of those, we're going to have to find a drug that operates. And it's going to be essential, one, to do the genome as early as possible in your life, because some of those variants cause disease during childhood or infancy. So you want to know immediately what kind of gene defects you have. But number two, our pharma industry is not poised to be able to generate all the different drugs that are going to be necessary to attack 7,000 different diseases with 7,000 different gene variants and things like that. So and it's going to be essential we do this on an individual basis, because the rare diseases occur once in 200,000 people. So we'll want to have genome-wide analyses of the whole population that, hopefully at birth can say, you have 4 gene defects you're going to have to worry about during your lifetime. And here are some that we have drugs for, and the rest we don't. And of course, it's your physician that'll have these data and keep track of these things for you, hopefully.
I guess for the pharmaceutical company's incentive to stay on top of this, what would be the financial model for how they can make money? In 30 years, you're going to get this disease, but there's a huge population and some percentage, so it's worth it for them to develop the drug or not.
I think the really key thing is to be able, one, to generate lots of new drugs that can attack more than 5% of the proteins that exist in your genome. The small molecule drugs today can only attack 5% out of 100%. So, that's a very small number. I think number two, they have to figure out how to scale the rapidity with which clinical trials can be carried out. Because if the clinical trial is going to cost $3 billion per trial, it's never going to work for these rare diseases. You'll never make your money back. So you have to have really efficient ways of generating lots of drugs, really efficient ways of screening lots of drugs effectively, and so forth. And these are things that pharma is just barely beginning to think about at a scale that's going to be appropriate for all the information that's going to come from your genome and your possible genetic defects.
Mm-hmm.
And of course, the question is really interesting. If you have a defect and can't do anything about it, one solution might be that only your doctor knows about that until there's an appropriate answer to the defect. My own feeling about this is I think knowledge is power, and I think everyone should want to know their beneficial attributes as well as their defective attributes and deal efficiently with both.
Hey, I just thought of a model. This print on demand, like, there's companies that make a lot of money, like print on demand T-shirts, right? But there's enough people that want that they can make a lot of money. Maybe pharmaceutical companies could be like print on demand drugs for this particular kind of cancer, for that particular genome. And there's enough people that need that they can make enough money cumulatively. So it's a slightly different financial model.
Yeah. Well, and I think any company that has the ability to generate lots of drugs and screen them effectively could line up as that for that as a possibility. And
right.
I think in the end, too, the federal government's probably going to have to help financially. I mean, if you think about a disease that's devastating in infancy, you've wiped out the productivity of that person for their life. And if you can avoid that, you have a productive, creative, functional citizen. So you can make compelling arguments about the lifetime value being able to deal with these diseases at an appropriate time.
Certainly, the U.S. government does this all the time. They subsidize industries, infant industries, to get them started. The example of the electrification of the rural America in the 1930s, this wasn't going to happen by free market capitalism, right? Roosevelt basically had to go in there and subsidize the electric companies who could not justify running a whole line down for one guy. There's a farm at the end of the road, right? So, but this is good for the nation, or the internet is the other big example. This is good. We should do this. And so this would be an example of that.
Well, another example that follows right along with our discussion here is the idea of data deserts for certain physiologic conditions. So I would argue, at least up until recently, pregnancy and what happens during that 9-month period is an enormous data desert. Very little detailed information is being carried out, but Nathan and I, together with Yoel Yasovsky at Pittsburgh, are now carrying out a 200-person project on pregnancy, looking at this enormous data all the way across the 9 months of this thing. And we would argue that the information coming from this will probably exceed the information we've learned in all of the past scientific history. But I would argue other data deserts are early childhood development. We know almost nothing about how kids develop. And I think menstruation is an enormous data desert, which has devastating consequences for women if it's not dealt with in an appropriate kind of fashion. So there are places where there are real imperatives to start looking at this big data view of physiologic conditions like pregnancy or menstruation, or disease conditions like rare diseases.
Interesting. Nathan?
Yeah, I think that there's just a tremendous amount that we can push forward and learn in these areas that we that have been not as studied, and that we can really get into in a lot more detail. So one of the things that's really exciting now is how we can deploy these new measurement technologies and how we can get access to information just in a much easier way than has been before possible. So one of the things that we've looked at a lot is being able to deploy remote blood measuring devices that you can mail to a person's house. You can put it, they can self-administer it, get access to a blood sample. You can pull it off, you can put it into the mail, ship it off. And what this allows us to do is to get access to information in ways that is simpler and cheaper than we've been able to do before. And so we are looking at this across large numbers of studies in many of these areas that have been historically less studied, and get into that in really important ways, I think. And we do have a device on that called the OneDraw, which helps do those kind of things.
Right. So you're really at this intersection between academia and private industry and government to make that transition from raw knowledge to application to an industrial size production of these kits and applications and so forth. I just had Michio Kaku on the podcast talking about quantum computing is going to be the next big revolution in medicine everywhere. You'll just have some device in your toilet every time you pee or poop. It tells you it gives you an update. I mean, this is astonishing. But how long will it take a company like yours to, make that cheap and available for everybody? When's this going to happen?
Yeah, so we've already talked about some of the things that are deployed, like the microbiome test and so forth. The blood measurement device that we just talked about, OneDraw, is approved already for direct-to-consumer use in many countries around the world. We hope that it's going to be approved very soon in the United States. It's already FDA approved in the United States for supervised draw, so you need a coach or some person to stand next to you while you do it at the moment. But we have successfully done this now in trials, including a big trial with the University of Cambridge on nearly 20,000 of these so far, with no reported adverse events and a 99.9% success rate at being able to get a measurement off the device when they do it at home. So we
How much
think
blood
that
do you need?
It takes 150 microliters of blood, so it's not very much. So it goes onto your
like those
150
little test
microliters.
tubes?
It's like a fraction of a test tube. So one of those test tubes is 1,000 microliters. We do 150, so it's about a fifth of one of the tubes that you would see typically in a
Okay, hang
blood
on.
draw.
In the light of Elizabeth Holmes and Theranos, how are you able to do this?
Yeah, excellent question. And I was pretty much obsessed with Elizabeth Holmes's story. I think I watched that documentary 20 times and read the book and the podcast. And one of the co-founders of Longevity, where I was CEO before Thorne, actually was one of the academics who did the first work on Elizabeth Holmes. Actually, I think his published papers is Joel Dudley. I think his paper is the only published paper with Theranos data in it, I believe. He did that right before the downfall, was a little kind of involved in that. So so it's a great question. So there's two different paths you can take. So one path is that you can try to get conventional clinical lab tests, which are typically done on large volumes of blood, and you can try to miniaturize that. That's what Theranos tried to do. And they failed, of course, and in fact were fraudulent all along the way, which is why Elizabeth Holmes is in jail. There are other companies that have tried to go down that route, and they've pretty much all failed. So that has been a very tough road to hoe. But there's another path, which is the one that we're pursuing, which is where you use small volumes of blood to do what are called omics-based measures. So these are your metabolomics, which is measuring all the metabolites out of the blood, or proteomics, which is measuring all the proteins. And those technologies, which are based on things like mass spectrometry or capture agents, are only ever done on small volumes. There's no such thing as running mils of blood through a mass spectrometer. Like, you wouldn't do that. You would. So these technologies only measure out of small volumes. We don't need any more. If we had a bunch of volumes, we would just take that amount out of it anyway. And so on that front, you have a different challenge. There's always a challenge. But so if you go down that path, then the challenge there is more informational, because now what we have is a new kind of set of markers that's much larger in scale. That small volume, we can make thousands of measurements off that. We just announced a partnership with Arome Science, which is a metabolomics company that's going to do our back end of this. And so we can make thousands of measurements off of that volume. Now the challenge now is that we have to be able to interpret all of that information. But with modern technologies, that becomes much more of a challenge of generating enough of that data, tying it to health outcomes, connecting it into EHRs, monitoring health in people, building out scores, knowledge. But it becomes much more an AI data problem to say, okay, with the measurements we can make at very small volume and taking signal that might be dispersed across a large number of measurements instead of one measure like we typically do in clinical labs. That's the difference in the challenge. So it's not a problem to do the small measures on that route. It becomes an informatics challenge. And I'd much rather try to solve the information challenge than to go down the road that people have died on many times before.
Can I ask you just a sidebar question about Elizabeth Holmes?
Let me just make one
statement. Please. No, go ahead. Yeah.
I am totally convinced that what Elizabeth Holmes projected is going to be done. I think just technically, as Nathan said, it's far more difficult. But we are going to learn how to, from a drop of blood, make 5,000 measurements, although it's going to take 10 or 15 years rather than 1 year, as she proposed, and everything. So I just want to say that is a valid way of thinking about the technology. And it's just a matter of getting appropriate miniaturized measurements in microfluidics or nanotechnology that will enable this. And my vision is in a 10 to 15 year time, you'll have in your home a tricorder equivalent that'll take a drop of your blood and make 5,000 measurements that we need to do all the relevant kind of things we're talking about here.
Amazing. Yes. Well, okay. So where does the fraud come in? Back to Elizabeth Holmes. Just because I'm curious, I don't know much about the case. I saw the documentary, but only once, and I didn't read the book. All these tech guys are, pitching like crazy, and they're projecting the future. Oh, we're going to do this. We're going to do that. We're going to make $1 billion. It's going to be great. And they all exaggerate, fake it till you make it. This is the kind of that crazy bro culture in Silicon Valley. Steve Jobs, her hero, was, famous for this. So where does the fraud come in? That she was intentionally lying, saying that she had already done it when she hadn't? Was it something like that?
That's basically the case. She claimed that they could do things by her technology that they were actually being done by other commercial technologies that weren't miniaturized. Okay.
Yeah, that's a big part of it. And the other thing is that the fake it till you make it in tech is a lot more accepted than fake it till you make it in medicine. So some of the things they were doing is they were giving tests for sexually transmitted diseases, for example, and they were putting them on the marketplace, and they had accuracy rates like in the 60s in terms of percentage. So you can imagine the number of problems that come out from that. You get your test and it says, "Oh, I'm clean," and you have a 35% chance that it's false. So there was something, and those numbers were being hidden, right? Any health group would never put those things out given those kind of accuracies. That's what some of the whistleblowers were really worried about when they came out. And so there was a lot of misrepresentation of where they were at. There were some fraudulent claims about having military contracts and backing that they did not have, some of which really came out as kind of obvious to experts because they were claiming to be on certain types of helicopters, which the process for getting on those, because they weigh those things out to the ounce, like getting anything on there, and they were sort of cavalierly putting that they had done this. And so there was a lot of it would have just been posturing, except for when it started having demonstrable harm to patients. They don't let you fake it till you make it on patients. You can't do that.
That makes sense.
Michael, we're going to have to run, I'm afraid. There is a commitment at 12 that
okay. All right.
We're going to have to go to. So
both of you.
I don't have to.
Okay, it's all right, Lee.
You don't have to.
I'll continue with Nathan, but give me 5 minutes. Do you have to go right now?
I do have to go right now.
Okay, all right, yes.
12 o'clock.
Okay, no worries. We'll continue with Nathan, but thank you so much.
All the answers. Okay, take care. It was really enjoyable.
And by the way, let me just say, I really found your chapter on Alzheimer's just deeply moving. I mean, I cried. I felt so bad for you. What a horrible thing. We'll continue with talking about Alzheimer's, but you've got to solve this problem. This could be a huge problem for us baby boomers. It's coming.
Well, Nathan can talk about that. Yeah.
All right.
Cheers.
Yeah. So Nathan, just one more thought, since you've thought a lot about Elizabeth Holmes. I'm fascinated by, I study, cults and conspiracy theories, why people believe why do people fall for people, like the,
I'm very familiar with that, by the way. I love all that stuff from you.
Oh, thank you.
We could talk about that on a personal level at some point. It made a big difference in my life, actually.
Like the Tinder swindler or the Nexium cult, these women that, had this guy's initials, branded in their crotch and all that stuff. But, really, they were joining, women's empowerment groups in business. Well, those are pretty common, so it doesn't happen quickly, right? And Elizabeth Holmes, there was something appealing about her, that kind of Steve Jobs look, the voice. Why did, George Shultz and all these, major heavy hitters kind of fell for that? What is your sense of what her appeal was, as opposed to like just some geeky guy that was doing something in Silicon Valley?
Yeah, And honestly speaking, that's a large reason why I was so obsessed with the case and like reading it and listening to it and watching the documentary so much was because of that aspect. There's also the aspect that it affects what I do professionally, but that was the thing that was so striking because I do think there's just something to she'd always wear the black turtleneck, right? So she's the female Steve Jobs. And in our culture, for lots of good reasons, right? We're so inclined to want to help the young female founder, right? We want to help we want to be the good person that helps the young female founder. And she had passion and charisma for sure. So she's got the turtleneck. Then she has that thing she does where she makes her voice low. And and you could see these things on YouTube, right? When she accidentally slips into the higher voice every once in a while, not very often, but every once in a while. And so it's very studied, right? Which is we do tend to find lower voices more authoritative. And that gets into all the issues that we do when we look at sex differences and so forth, and that a lower voice. And so she very clearly studies that. Then she's got those really big blue eyes, and she almost doesn't blink, right? So she's got kind of this, like mesmerizing aspect of just the big blue eyes, the lower voice, and the confidence, right? She has tons of confidence and charisma. And it is interesting, especially when you go through the history and the documentary and so forth. So all of her board members are older men, all of them, 100%, right? There's no women on there. In fact, they do an analysis of, or not an analysis, they do an interview with a senior faculty member, might have even been a dean, anyway, high level at Stanford, female professor that Elizabeth pitched at the early on, and she came away like, there wasn't anything. Yeah, she's nice. I like her, but like, what? Doesn't have a lot of technical background. Like just a very like reasonable analysis of wait, she's just finished her freshman year, and I don't think that she has solved this technical problem that no
but
one in the world has
but the
been able to
guys are
solve
like,
it.
but the blue eyes.
And exactly. And I don't, and it's not that simple, of course. They're like that's too simplistic, but there's something to, and I think the feeling that in an older man like there's a story you want to believe in, right? You want to believe you're not imputing bad motives. They've done sociologic studies you've probably seen on these, of people like go into a park and steal a bike, and if you're a black man, people are on you, and if you're a white man, they're kind of giving you a hard time, but they're not on you as much. If you're a beautiful young woman, they'll help you, steal the bike.
Let me help you put that in your car.
Yeah,
Yeah.
exactly. You've seen these like hidden video
Yeah.
things they do on this stuff, and so there's always like funny. So I do think there was some sociologic aspect of wanting to believe. She's also very well connected, right? She's in the right family. She's at Stanford, right? She's in the right family. She's at Stanford. You feel inclined to help her. You want to. And by that point, you've already bought in. You're doing it. And all the people she went to were not science experts, so they don't know the technicalities of what she's talking about. And she does have a force of career. I mean, there's a reason she was on the cover of Forbes as the youngest female turned out to be House of Cards, but the youngest female billionaire we thought at the time. And and so it was striking. One thing I will say though is a really prominent venture capitalist who was actually one of the investors in Aravel, and we were at a dinner party and was chatting with him. This was maybe a year and a half before the fall, and Elizabeth Holmes came up, and I said something like, I don't know, just making chit chat, right? Like, oh yeah, she's off, she's doing very well for her, I don't know, some offhand comment that got it started. And it was interesting because he immediately retorted and he said that I will bet you anything that she will be worth nothing. He says there is nothing there. And so when it all came down crashing down a year later or year and a half later, or whatever it was, yeah, it increased even my already high level of respect for him because he was absolutely right. They had done due diligence, and that was his take, that they had never shown him anything under the hood, and he was quite convinced. So there were these murmurs that were out there over time, but there was enough of people who did believe that if you on the surface, with Forbes magazine and all the things that had been going on, that that you thought they must have something. And in fact, I was talking about this in an interview actually, even earlier today, just came up randomly. But it was she came to give this big talk at the American Association for Clinical Chemistry, AACC, big scientific group. It's all the people that make blood measures, the super experts. They're all at this meeting. It's very hardcore science. And she was giving a keynote. And the fall after Joel's paper and a few other things was happening. So she was on the rocks. She maybe didn't understand how badly it was, but she was giving this keynote that she'd been invited to. So it was all this drama that had built up. And I was watching with tons of interest, like everybody, right? Okay, Elizabeth Holmes, she's giving the keynote. She's going to show something. It's put up or shut up because her company's about to get decimated. And she got up and started talking and giving this keynote. And it was a total pivot to, I'm going to build this different device that doesn't exist. And about 10 minutes in, I had to pick my jaw for the floor, and like everyone, because we're looking at her like, he has nothing. Because if she did, this would be the moment to show some data and say, this is all nonsense. Like, look, here, even hiding whatever proprietary, you would say, look, here's something happening. And it was a stunning moment. And it was
wow.
Where all of a sudden you realized, oh, there's nothing there. Absolutely nothing. And then then everything else cascaded out afterwards, right? And we learned all the details, and you just thought, wow, what a mess.
Well, on the question of retributive versus restorative justice, this is legally way beyond you and my expertise, but so she's going to prison. She owes $100 and something million. How are they going to collect if she's in prison? I mean, I don't know. It's like maybe leave her out. It's punishment, I guess, for fraud and a signal to other people, don't do this. But what if she was freed to work and they just garnish her wages and just start collecting? I don't know. I don't know who she owes the money to. I guess the investors.
Yeah, it gets to be an issue. Yeah, and we are getting beyond our expertise, but the one thing that I would say in this case, the investors that put money into her didn't do their jobs. I mean, they never did enough scientific due diligence, for sure. So, and losing money when you don't do your scientific due diligence is a pretty expected outcome. Like, you're going to have to live with that. And so I do think they have made her an example. The cards could have fallen differently where she does, probably doesn't cascade this badly for her. And it is a little bit of a chilling effect on the field, because every time you say, hey, we can make a bunch of measures out of a small droplet of blood, of course people say Theranos. Of course you would. Who wouldn't? Mm-hmm. Even though you might be talking about a technology that's radically different, and there are all kinds of ways to pull it out, but you have to be careful. And the good thing about it is that they do push more scientific due diligence now. Like, you have to show, which is
Well, especially for venture capitalists, they have to, because most of the companies they fund are going to fail. Like, what's the the back of the envelope calculation? You hear 100 pitches, you fund one of them with millions of dollars, and then of the 100 that you fund, maybe one becomes an IPO or gets bought by Google or Apple or whatever, and then everybody makes their money. So it's mostly failure, or just nothing big happens. It just kind of maintains, right? So you'd have to be super skeptical. So that that would explain what so your analysis of her looks and voice and all that stuff would explain why they kind of dropped their skeptical shields there. Other cases, like I mentioned, the Tinder swindler. I watched that. If you haven't seen that documentary on Netflix, it's quite amusing. Because on the one hand, they make these women look like idiots that they gave this guy tens of thousands of dollars and more in some cases. But the build up to where he gets these women to give him money, right? I mean, he's on Tinder. They look at the profiles. They know a lot of the guys are bullshitting them. And here's this guy standing next to a private jet. It's like, yeah, okay, maybe he's a good-looking guy. I'll swipe right or left, whichever the one is where you say yes. Yeah, right. And okay, I'll go for one date with them. And they're on the plane, and they go to this island and there's a 5-star resort, and the whole thing's super romantic. And then another one and another one. It's like, okay, and then and the women have like thousands of texts from the guy. You're going to have my baby. We're going to build a family. It's going to be great. You're the one. And these women are all in their like mid to late 30s, and they, this is it. I want to get married, have a kid, start a family. I'm at that point in my life. This is the guy. Oh my God, I can't believe I hit the jackpot. and then the moment comes where, he's an international man of mystery and he goes to Morocco or somewhere, it's like, oh, darling, I have to wire this guy $10,000 and the bank's closed today. It doesn't open till Monday. I got $10 million. The bank is no problem. And at that moment, she's built up to this. Do you say no? Like you're turning down the guy you're about to marry and have his kid? Right. So some of them said yes. Now, what we don't know is the base rate. Like how many people did he try this on and it didn't work? Right. And of course, they're only highlighting on the show the ones that fell for it. So I'm always curious about this, default to truth theory. Do we tend to trust people because who has time to fact check everything? Now, your investors, for Elizabeth Holmes, they should fact check. But the average person, when they're just telling you stories, who has time to check everything, right?
Right. Well, and especially once Elizabeth got it going, right? So once she has the mythos and she's got those people, because you think, all right, I got to do my due diligence, but you look and you say, he has the Secretary of Defense, right? You think you start having, because like you say, like reasons to believe, right? Which is something I think about a lot too, because it's such a fascinating topic. So it's really hard to not believe things that you really want to be true. That's a first thing. And then it's really hard to not believe things that you've invested in for a while, right? Because you remember Secretary Schultz, like it was his own, I think it was his grandson, that was one of the whistleblower, and he was taking Elizabeth Holmes' side over his grandside initially, right? He flipped eventually, but he had already, because you've already invested that much, and there's a myth, and so once you have a set, you just think, oh, it's got to be true because there's so many other people, all the other important people believe it, and you start getting into this whole cascade. And it's one of the famous Richard Feynman quote that so many of us love, but that the first thing is not to fool yourself, and you're the easiest person to fool. And you have to think about like, here's all the reasons, and it's true in science too. If you get too down a hypothesis, right, and you've written a bunch of papers, you believe in something, and then contra evidence comes down. And you shouldn't always change your mind every time every piece of contra evidence comes down because you'd be too wishy-washy, but it should give you pause. And you should know that if your career is based on something being true, all of a sudden you're the super expert, but now you have more invested. So you're now the least unbiased person that exists to determine whether or not a new piece of evidence changes your ideas. And all these things are incredibly challenging in both professional and personal life, of course, to just think through, what are beliefs that you have that you should reconsider and give up. And that's a hard thing.
And back to your field, I mean, the replication crisis that started in 2008, that spread through psychology, but also through medicine, because a lot of these experiments are really complex and hard to do. And so the peer reviewers who we trust, how closely can they get to the data to see what was actually done? If there's a file drawer problem where the experiments that were run that resulted in no results aren't even reported, how would a peer reviewer know that? And most of them don't see the raw data, right? So,
right,
who do you trust?
Yeah, one of my professors at UCSD, one of the things he drilled into us a lot at the first year, and I really liked this, was, the peer-reviewed literature is not the truth. Peer-reviewed literature is the average. And that is exactly true. And I've peer reviewed hundreds of papers. I've had hundreds of papers peer reviewed, and so been on all sides of this process. Been an editor of journal, all that stuff. So all over this, and peer review is important. Like it's helpful, but there's no way that you can absolutely identify the correctness of everything in there. And in fact, I served on a few committees for the National Academies and the National Cancer Institute, where we looked at this issue of data reproducibility, and it's incredibly hard. So in the early days, when you started being able to make lots of measures, you could measure thousands of measures on what's called the transcriptome, so the expression of genes and so forth. And there were different algorithms that would determine how you go from looking at, say, a spot to calling a number. And when we did careful studies of this, you could compare like cancer versus normal. That's a huge biological difference. But if you looked at the different computational algorithm that was chosen on how to, which is an arbitrary detail mostly left out of papers back then, that decision was larger than the difference between cancer and normal. And we did all these analyses where we'd say, which is easier, to predict the lab who made the measurement or the phenotype, whether it was cancer or not. It turns out it's easier to predict the lab that made the measurement. There's more variance captured there. And we did a whole slew of papers on this back in the day. It was a hot topic at the time. But it gets into all these issues. And so then when you go into peer review, the other hidden issue, and we got into this in a National Academy report we wrote, that I was a participant in, that we did some years ago. But as you get into this, there are one of the big issues is if you try a whole bunch of things and then you only report the one that worked, right? It's all this there's hidden multiple hypothesis correction rampant across the literature. Because, and this is why in my lab, I always tell my students or postdocs when we start something, there's two practices we do. One is that we take 20% of the data and we wall it off and we throw it into a closet that you can't look at. And you can't look at it till the very, very end, till the week before we're going to submit the paper. You can't look at it because to hide for this, right? Because you're going to try some things and you're going to do a bunch of different analyses, and that has to be naive to all of it. You have to wall it off. So we do that. And then the other thing I tell them, which is a little counterintuitive, I tell them, don't try too hard. Like, we're going to try this, we're going to come up with this analysis, and we're going to see how it comes out. But don't bang on it and tweak it and change it and try a million parameters for the next few months. Don't do that, because then I know for a fact that when it comes back, for sure it will die when we open the closet. And so those are, it just does. But that's not enforceable at all because it's invisible to peer review. Peer review only knows what you point to at the end. They can see if you did the statistics at the end right. They have no visibility into what you did before, and the whole game is there.
Yeah, there's this push to post in psychology, anyway, to post your proposed research online and everything you're going to do. And so it's there. There it is. Okay, you did 9 experiments. All right, we heard about the one. What about the other 8?
Yeah, that's a proper way to do it. It's a great way to do it. It's a little hard to do if your analysis is really exploratory.
Oh,
that makes it a little bit harder, which is why I don't do it entirely that way. But if it's a more straightforward analysis, if it's an analysis of a kind where you're not inventing some new method or something like that, then 100% that's the way to do it. And that's why we require registration of clinical trials in advance, because that's when it gets serious. It says, no, you're going to run these analyses, and you say in advance what the criteria is. You can't fuddle with it after the fact.
You have a sense of, what's come down after the pandemic. You can't trust Anthony Fauci, the CDC, all these medical researchers. They say masks don't work. They do work. The vaccines will stop the spread. It won't stop the spread. I don't know what to believe. Ivermectin, no. Ivermectin, try this other thing. Well, I don't know what, There's been a collapse in trust in the medical system because of the pandemic. What are your thoughts on that?
I think one of the first things I really think is it's an introduction to how hard it is to know definitive answers to simple questions. Because the time it takes for us to really come up with definitive answers is typically long. And the answers, when we call them definitive answers, are usually a lot less definitive than we think they are. And there's super outliers, things that we understand causally, but the broader you get, because you would think something simple like, should you wear a mask? Right? That seems pretty simple. And there are arguments that I've heard people say in both directions that say it's super obvious. One of the big things that it does for sure is it limits bulk airflow, right? So your airflow is not going to go around. But if you're in a tightly confined space with somebody, for a long enough period of time, that variable won't matter really anymore because the virus will circulate and it will get in, and you have enough gaps. Unless the thing isn't incredibly tight, it's not going to hold out the virus. It's so small, and so it becomes an issue. But if you're going to go through for a more limited period of time in a certain space, and that bulk airflow matters, then sure, it should help. Anyway, if you're outside, presumably it doesn't matter because the density, because I saw so many people, right, you're super masked up outside in the early days, and you're like, and they give you a bad look if you weren't, but you're like, the data is like, well, we're outside. So this bulk airflow is not going to matter. And so, and I wouldn't purport for the same reason that it's been complicated for everyone else that, that I have the answers or that I'm the super expert on this or anything. But I just think these things are hard. And the other thing that it really showcases is how much easier it is to be right in retrospect than prospectively. And I'm, well, there's this, I don't know if you ever watched South Park, but there's a character on South Park that I just think is hilarious, who is Captain Hindsight, right?
Oh,
Captain,
yeah.
have you seen this? Like
Yeah.
Captain
Yeah.
Hindsight, where he'll fly into any disaster and tell you what you should have done. I
That's
just
really
think
funny.
I think that is just so brilliant. And I just love that. And so every time I watch the news or so after some huge event and you have your experts come on and tell you what you should have done, I just
Yeah.
think, Captain Hindsight coming in. So there is that element of, the people who are in the seat that really mattered at the time, they're just under an incredible amount of pressure that those of us that want to armchair quarterback afterwards are just not even remotely in the situation that they were in. But it is important, that joke aside, it is important for us to look back and try to learn lessons from it. And there were definitely places where we got overly dogmatic. And I think there was such a move to not, to try to save lives, but there was a lot of shutting down of what I think are very reasonable things to talk. Like, for example, the lab leak hypothesis, which is not crazy, right? I mean, it could.
No, of
I
course. It's
I'm
totally
pretty
reasonable.
I'm pretty inclined to think it's the more reasonable, more
Yeah,
likely outcome.
me too.
But they banned people right off of social media. You
Unbelievable.
would cancel for that, right? And I thought even at the time I thought that was ridiculous because I like to consider myself more on the free speech absolutist side of things. But that was a yeah, and so we know that there was a lot of overreaching where we got into where you couldn't have a reasonable debate about things that mattered. And the other thing that was really disheartening, I think, for all of us was how much if I asked, if you knew how you voted in the last presidential election, I could say what your opinion is of a mask. Those things should not be correlated.
No.
And there's of course a
Yeah,
million examples like that. I really, I don't like strong correlation across ideas that have nothing to do with each other. I think
yeah.
those are bad social phenomena.
And on the vaccine and the spread of COVID, so the argument is that the vaccine helps your body fight against the virus, so your symptoms are attenuated somewhat and last fewer days or whatever. So the
Right.
idea would be if I'm going about my day, I'm less likely to sneeze and cough and so on. So there's fewer droplets going out to spread or something like that. That would be the argument, right?
Yeah. And I think it's a reasonable argument. And again, not core expertise, so people could take it for what it's worth. But the data that I've seen most recently, it seemed to suggest, at least what I've looked at, that the vaccine didn't really help much in terms of spread.
Right.
Helpful for reducing your symptoms. There was hope that it would really reduce spread. Um, I think the results on that are pretty mixed
Yeah, that's what I've seen too.
from what I can see.
So here's how I see the problem. It's a signal detection problem. So picture our 2x2 grid that I always use. So in this cell up here, you have that the COVID-19 is going to be, let's just say, not that bad. And you're the public official and you say it's not that bad. So that's a hit. That's correct. Or it's going to be horrible and we shut everything down. So that's good. That's another hit. And then the two errors, though, are that it's going to be horrible, and you say, let's just carry on business as usual, and 50 million Americans die, or even 20 million or something instead of 1
Right.
million.
Yeah.
That's on you, right? So better to make the other kind of error. Let's assume it's really going to be bad, and it turns out it's not that bad. Well, as we did was shut down the economy and close schools and so on. What's the harm? Well, okay. So in other words, you have to kind of balance out those two kinds of errors. So I try to picture myself, I'm the guy, the mayor, the governor, the president, and they stick the microphone in your face. What do we do tomorrow? Do we close the restaurants and schools tomorrow or leave them open? Go. And it's like, fuck, if 50 million people die, that's on me. So I better say the other thing.
Of course, of course. I mean, I feel for them, right? I mean, what a choice. And you're hated either way, for sure.
Right.
You know, a bunch of people, you don't shut down and people die. No matter what the number is, it's your fault. And there's no counterexample. There's no counter running the country in a different way. So, so that's an issue. And what that highlights, though, is it brings to the fore how much of an issue this is for other diseases, but it's hidden. We don't think about them. But we regulate a lot in medicine. And one of the results of that, so partly is for safety, and there's all kinds of reasons that that's a good thing. But what's hidden is that we move the health. So there was an analysis done across many industries about the pace of technological adoption and efficacy and efficiency over the course of the last few decades. And as you would imagine, the tech industry crushes on that. It's better and cheaper exponentially every year. It's amazing. It's created insane wealth. Medicine, though, is one of the slowest moving technological adopters that we have. And it has become less efficient and more costly over the last 3 decades. And that shocked me. And they did a head-to-head comparison against a really new, fast-moving, just brand new to us, a sector that was crushing medicine on that regard. That's called farming. I don't know if you've heard of that. So farming, farming, which we've been doing for hundreds and hundreds of years, or it's deathly old, but has been advancing at a faster rate on these large metrics than medicine has. So what we don't ever talk about, because regulators are in exactly the place you talked about, they approve something and it doesn't work, they're in trouble. But if they don't approve it, and unbeknownst to us, 1 million more people die, that's on nobody. It's just the world. And so the cost benefit to a regulator is not the same cost benefit as to society. And one of the things we saw is that we did accelerate a bunch of stuff in COVID, and we showed that we could get things done much more quickly than we typically do. So if you look at Alzheimer's or cancers or different things, there's so much drag on the system to push things forward. And I'm not talking about not having drag on things that are obvious, like a new experimental drug. But if you want to do something that involves, brain training and a blood test, or let's say, which is, computer games and a blood draw, you still got to go through a lot of red tape and a lot. You might spend, you'll spend as much time working through the red tape of doing that as you will working through the science of it. And that's to do something where the danger level is very, very small. And so, and I serve on some of these safety boards and things like, so I understand, and we work within the system and so forth. But there is this interesting dichotomy, and I know regulators think about this too, where there's a hidden cost of slowing down, and there's a not hidden cost on the other side. And so we're very biased towards more and more stasis if you go down that road, and if you never shake it up, it's a huge issue, and it's hard. It's a hard one to advocate for, because if you advocate for it too strongly, it sounds like you're going against safety regulations or something like that, or you're against institutions that have enormous power to destroy your career and all those kind of things as well. So but it does create a toll on society. And one of the interesting things is, because if we look at mice, right, we look at mice where these don't exist, and that's an even more dangerous argument to make, because obviously there are reasons we don't do in us what we do in mice. But the one side I want to point out for that is there's tons of diseases that we can cure in mice, no problem, because we understand them, right? And so there's got to be some medium, something where we can think about how we enable ourselves to have the best way forward in health. And I do think a lot of that coming back to the book and everything on the age of scientific wellness. I think part of that is to try to have a focus on how we extend health span, how we have interventions that are largely safe, natural products, lifestyle, dietary, to the extent possible, things that we can deploy that are not so dangerous, and that we can have avenues where we can push those out quickly, efficiently, take advantage of AIs and the measurement technology so we can learn a ton from the human population as a function of us just going through our days and our lifetimes, and that should create this major revolution in how we can optimize health over time. Anyway, those get into some difficult issues, but those are kind of how I think about it.
Yeah, no, I was thinking about that, not doing something and the hidden costs of that with that call for a 6-month moratorium on development of AI because of ChatGPT, just in case, at least to the extinction of the world, extinction of the species and the end of the world as we know it.
Right.
It's like I've heard that before, many times before, and it never happens. But, what about AI developing to analyze Alzheimer's and cancer? Maybe we should not worry about it writing and passing the bar and hoping can, make some advances in Alzheimer's. That just drives me crazy. What do you mean pause technology? Okay. All right. So there's no
chance. There's no chance that will happen.
Well, it's not going to happen anyway because certainly the Chinese and Google and Apple know.
You can't do it.
So there's no Moore's law. There's no Ray Kurzweil like singularity exponential growth in medicine. Why not? Is that because it's overly regulated? Like why do we have a Moore's law for nuclear energy? Because it's overly regulated. My sense is medicine, it's not like that, maybe because the problems are way harder to solve, or maybe it's both.
Yeah, it's a great question. I do think part of it is certainly regulation, and part of it is just the sensitivity of progress in medicine. And I don't want to come across as like I'm negative on all that. I'm definitely not. A lot of it is really necessary, but the big issue is the failure of experimentation, at least in some cases in medicine, is pretty catastrophic. This is why we don't have exponential increase in gene therapy. Gene therapy, incredibly promising, incredibly dangerous. Stem cell therapy, incredibly promising, also dangerous. You have, so there's definitely a slowdown in terms of just inherently the challenges in medicine, and that we are biased so strongly towards do no harm, right? We want to.
Okay, so but what are the dangers of those two?
Gene therapy? Oh, yeah. So gene therapy, right, with CRISPR, right, which won the Nobel Prize, very deservedly so for the. But basically, if you look at CRISPR, so CRISPR allows you to go into a genome and edit essentially any base pairs that you want to, the first approximation. So it is incredibly powerful, and it holds the potential to end all genetic related disease. Or at least like monogenetic defects and things like this, so you can go in and change. So from that standpoint, it has the potential to be pretty miraculous. So if you're going to get Huntington's disease, you have an extension of the CAG repeats that extend out in that gene. And Huntington's is a particularly compelling example because unlike most genetic traits, if you have the gene for Huntington's, you will get Huntington's disease in your life. It has essentially 100% penetrance. You get it. But you can CRISPR it out. Now you can get rid of it even in embryonic stage, and you can do this also just through in vitro fertilization and selection. Actually, a friend of mine did that. He has a whole documentary on himself about all this because his whole family history is Huntington's, and then he eliminates the gene, and all of his progeny forever will not have Huntington's. That's if you're able to step back and think about the scope of history, for 100,000 years, who knows, thousands of years they've had it, and now they won't. Amazing. And we can do that very broadly. Now, the danger of it is, and this gets back to our conversation about systems biology before. So we know the genome, and we know we can change it. So at the molecular level, it's solved. Reductionist, it's solved. But the systems level is not solved because there are unintended consequences. Because what we don't know, especially when we start dealing with complex disease where there's multiple factors, is, okay, you go in and you change, like let's say you have ApoE4, it gives you higher risk for Alzheimer's. All right, let's go in and change it to ApoE3 or ApoE2, protects you from Alzheimer's. Let's do that. Okay. but there's probably a reason that ApoE4 evolved. And maybe in the context of your genome, there's some interaction that's going to become problematic for you with this, with this other copy of the gene. Or maybe you get, or maybe it relates to some other trait you have. Maybe it has an influence on your intelligence, or maybe it has an influence on your personality. Maybe it has, we don't know. And the problem is that we have so many unknowns. So when you come in and you start using this incredibly powerful technology, saying, okay, because in theory, we could go through our genomes and we could say, all right, here's all the disease-causing genes, make them all the good ones, turn them all to the good ones. And you do that. But the problem is that it's not one gene, one trait. These are all complex systems. They come together, and we're going to find that there are, and the first trials in gene therapy, this is what happened. Like they had horrible side effects, you get it messed up. And so the problem is, can we be smart enough? Now, this gets back to the other point you were making, though, about AI. And in fact, I think AI is essential to us figuring out how to really deal with systems biology and the complexity of biology. I think without AI, we either never get there because we just can't get our minds conceptually around everything, or we have to build tools to do it, right? And AI is clearly the next tool. And so,
I think this is an example of pleiotropy you're talking about, where genes code for many different things and interactions. So my favorite example of that is those silver foxes in Russia, where they bred for docility, where you just measure the most tame one that will come sit in your lap or whatever, and then you just breed them. Pretty soon you got these super docile foxes that also have the curly tails and the star-shaped thing and the floppy ears, and so you're actually breeding for a whole bunch of things, and you had no idea that it was going to go that way.
Exactly. And slightly different topic, but this is one of why the breeding of dogs and so forth is one of the most just potent evidences for evolution, right? You take them, you cross them, and you see over the course of a very short period of time the incredible diversity that we see in dogs. And yet I was just looking into this the other day. The genetic diversity amongst dogs is not that high, right? They haven't been around that long. It's not that divergent. And yet the phenotype is
unbelievable.
Unbelievable. And because we forced it a little bit, right? It's just, it's evolution in action. You can see it. It's really remarkable. Now, if we really understood all these systems, we should be able to print a genome to create whatever organism we want to. Now, that is a capability we are very far from having. And we're so far away because the complexity that comes after the genome and how all these pieces fit together, we only very vaguely understand. And that's not to put a slight on any of the science that we've done and other people have done, but it's complex, very elegant, great papers, beautiful science. But compared to that challenge, right, that we actually understand everything from a genome to be able to create the organism that comes out, we're way, way far from that. And so those are the issues that you get into, though, when you think about gene therapy, stem cells, being able to regrow the brain's synapses following Alzheimer's disease, like being able to actually regress that fully back. You could imagine ways to do that, but they're totally sci-fi today. And this gets again back into what we talk about in the age of scientific wellness, because if you think about something like Alzheimer's, bringing your brain cells back and your synapses back once they've died, nobody knows how to do that. But prevention, stopping your neurons from dying in the first place, is in a completely, totally different league of challenge. It's way, way, way more doable than regrowing a brain. There, all you have to do is figure out, okay, what is it that's killing off the cells and stop that or slow it down. And there's tons of things we can do that will make a difference on the delay of we've all known this since we were 3 years old, right? It's the story of Humpty Dumpty, basically. But but you can just stop. And so those are the things that we really want to get medicine much more focused around, which is if we focus on health and the extension of health span and the processes that lead us towards the negative outcomes that we don't want, right? The early deaths, the loss of of loved ones too early, all these different things. We have not spent anything like enough time and effort and science to deeply understand how is it that we extend health span, because we organize all of our scientific studies around diseases. We put all the money centered into disease, and so what we have created then is a disease-centric, after you have a serious problem, I'm going to give you a drug, and there's tons of money in that. But it has not actually worked very well for our modern issues of chronic disease. It worked fabulously well for infectious disease when we had something to target and kill. We did that beautifully, amazingly so. But chronic disease, we have not been that successful. And that's what we argue in the book largely, is that if we took this very different approach, much more centered on the extension of wellness and prevention and the deep science around that, that we would in fact get ourselves into an area where we could make massively more progress at slowing aging, stopping the progression of disease in its earliest stages, and not getting into the terrible situation downstream that often we're kind of fooling ourselves that we can reverse.
Yeah, I want to explore cancer and Alzheimer's a little bit more, but the second before that, the second point on stem cell danger. So before I had my titanium hips put in, I had people say, "Oh, you get the stem cell treatment." It's like, well, how does it work? Oh, they inject stem cells into the joint, and then they do what? Well, they rebuild the bone. How did they know how to do that? Well, then the answer I would get was like, and then a miracle happens. It's like, I don't know. And I read some about it, and I thought, I don't know. This doesn't look like terribly promising to me.
Yeah, it's kind of true. Stem cells are a little bit miraculous in a way, and I don't want to lump them in with gene therapy. I think is more dangerous. But when you're putting in stem cells, the stem cells are the cells, of course, that grow all the other cell types. So they are generative, and you can have what are called induced pluripotent stem cells as well, where you can, and so stem cells are an incredibly promising therapy, in part because they are so intelligent in a sense. Because your doctors are exactly right. Like you put these stem cells in, and they are programmed at some level to grow tissue. So they are very powerful in that sense. And so you put them in, you are taking a bit of a leap of faith in a way, because you are saying, because it is because you're injecting it into that site, and you are trusting to some degree in the intelligence of the stem cells to act on their programs and to rebuild cartilage in some sort. You're not totally taking on faith, because you're doing these experiments, right? You do experiments, you look at the data, you follow it up, you see that people do better on it. I mean, it's all evidence-based that way. But we don't understand all of the logic of how they do it. Right? And so those are cases, though, which is something I think is really important to the kind of where we're going with biology, though, which is how do we have a dialogue with some of our really sophisticated biological systems? How do we understand their language? How do we talk to it in a language it understands? And I don't want to anthropomorphize too much, but just try to give a view, because we have we'll get into it, especially when we talk about cancers, because when you get into immunotherapy, you're trying to harness the power of the body around its own immune system, and how do you augment it? How do you make it how do you help it to fill in gaps that it's missing? Right? And because most of your cancer, so all of us, if you live long enough, you get cancer, right? And we may have already had cancers already in our lives, but they started and our immune system cleared them out, right? Your immune system is clearing out cancerous cells all the time. But when you develop a tumor, it means that something went wrong. Your immune system didn't recognize it, and it didn't clear it, and now it's growing. And your immune system failed in that regard. And your immune system is incredible. And I forget the exact numbers. I have a friend that really did a lot of work on this at MIT, but you've got every time you try to do a classification problem, right, it's trying to determine what's you and what's not you. And the number of variables that it's actually having to distinguish between is astronomical, right? It's huge. And it does a really good job of that, but it's not perfect. It's probably not even possible to be perfect, at least in any system that we can conceive of. And so when it fails then to clear a cancer and it's growing, it means that that cancer has figured out how to fool the immune system, how to grow unchecked, how eventually how to metastasize to other parts of your body. It's going through that. So one of the things that's so exciting in the cancer field recently is the notion that you can do immunotherapies, which is essentially to not try to just kill the cancer off with a drug, but rather to say, can I teach the immune system that it missed this, that it should do it? One of the great pioneers in stem cells and in immunotherapy is Irv Weissman at Stanford. This is where Lee would be really helpful because Irv and Lee are old friends since high school in Montana. So they own a ranch house together. This ranch that we go out to every summer, they own it together, like it's their place. And Dave Baltimore, President of Caltech, former President of Caltech, has a little place down there, close. They all have an old Jeep that they own together, which is anyway.
It's funny.
The kind of thing like that, but he created a technology that creates something called an eat me signal. So what you can do is you can program. So if you know the molecular properties of these cells that have become cancerous that your immune system has missed, you can create essentially a vector that will go in and it will look for certain gene expression or molecular properties or some combinatoric aspect of those cells. And it will go in there and then it will initiate a program that will put a molecule, it'll stick a molecule up from the outside of the cell that is a signal to macrophages. So these cells in the immune system will come and eat other cells. That's essentially an eat me signal. And Irv turned this into a company and they went huge. They're worth a fortune now. But it's an incredible technology because it gives you an ability to start this dialogue with the immune system where you're essentially taking advantage of the fact that the immune system will eat cells that have this molecule stuck up because that's what it uses. And so all you have to do then is introduce your own program with its own logic circuit to recognize what you have done because you've sequenced and looked at the molecular properties of the tumor, have it flip this thing up, and then your immune system comes in and cleans it for you.
Is it a chemical signal that they look for in the bloodstream that these cells are giving off?
Yeah, I don't remember for sure, but I believe it's a protein that sticks up from the cell membrane that it recognizes and it clicks onto.
So the immune system cells, the T cells or whatever they are actually bumping into other cells and looking for the molecular.
It as the antigen, and then the antibodies will attach to it, and then the macrophage comes out.
I see. Okay, right. And then there's these stories about injecting tumors with vaccines. I think Jimmy Carter had that treated with his brain tumor. I think they injected it. How does that work?
Yeah, so it's a similar kind of idea, which is you are taking pieces of the cancer cells, and then you are creating you're creating antigens to get the immune system to create antibodies to it. And so when you have those antibodies, then it does the same thing. It will glob onto that. So it's the same sort of idea. You're giving a signal into the immune system that says these are fragments of something that's foreign. And then once it understands that that's foreign, it can then look for those tumor cells and kill it off. So it takes advantage of your immune system in really deep and incredibly promising ways. Like if you look at this, so it doesn't work yet for the majority of cancers. It works for a subset, but for the fraction of cancers where it works, it's amazing. Like you can look at, like someone will have on their back like all these lumps, right? And they'll start an immunotherapy, and 7 days later it's smooth and it's killed it off. And you can
7 days.
have these
Whoa.
7 days. And so you can get these responses. And it works especially well in liquid tumors. So, blood cancer, certain kinds of blood cancers, those are a little easier because you can pull out the blood, work with it, and then put it back. But as you do this, it becomes a very different style of problem. Because for so long in the war on cancer, we just didn't have anything to do about the cancer, right? We had marginal. I mean, chemotherapy is, it's helpful, but it's barbaric, right? You're killing every fast-growing cell, right? All your stomach lining is dying, your hair is falling off, and you're killing the fast-growing cancer cells. And it's a race. You're trying to say, well, the cancer will die before you, so we're going to go through this horrible thing, and then we'll come back. People in the future will view chemotherapy as barbaric. They'll look at it like we do leeches and stuff like that. And even now we understand that. It's just you survive better if you do it, but it's very non-ideal. But now we've transitioned into a world where there are really effective treatments for a small subset of cancers through immunotherapy. So now we have a very different problem, which is how do you take these therapies that work for 5% of people and make them work for 20, 50, 7,500? And the other big problem with some of these therapies is that they're super expensive. So maybe it's $1.5 million to go through and do it. And I'm not sure what the current, I know that's around where it came out at. Because you're doing all this N of 1 analysis, right? You have to you have to do all this deep molecular analysis of that person's tumor. You then have to pull out the pieces. You have to be able to program it and come in. But what I would, and there was a lot of angst about this when it came out at the time, and big problem. But I think we're in such a better place to have the problem being, okay, we have something that really works. How do we figure out how it works for more cancers, and how do we make it cheap? That's a way better problem to have than we don't have a clue how to treat cancers. And so I do think we've gone through a phase shift that hopefully we'll be able to make these things cheaper and cheaper and more expansive. And there's tons of people working on that right now. It's an incredibly hot area.
So part of the problem with cancer is that there's so many different kinds, and they're different in each body, to the point where you could say, if you get prostate cancer and I get prostate cancer, they're not the same cancer exactly. So the current medical model is, well, for every million men that have prostate cancer, this is the treatment we use, and it works 80% of the time or something. That would be the best. But what you're saying is that ideally, if you knew my genome and the genome of my prostate tumor, then it's mine. And there's nobody on earth that has exactly that, right? So you've taken a sample size and you've said, okay, all men that get prostate, all white guys that get prostate, all white guys that are 68 that get prostate, all white guys that are 68 that were once cyclists who now live in Santa Barbara. Pretty soon the sample size is one, namely me.
Exactly. And so this is why precision medicine is probably the most advanced in cancer compared to any other area. And it's because of exactly what you said, because you can pull out the tumor and you can sequence it. And you can look at its gene expression or its metabolites, its proteins, all kinds of things. But you can take your genome and the genome of your cancer, and you can identify all of the mutations, all the differences. So you actually have a map on a per person basis of exactly what's different about those cells. And so that becomes the basis then for designing a individualized therapy. Now, it's even more complicated than that because amongst your cancer cells, there are different subpopulations. They don't all have the same genetics. Some of them are different. And that's the big problem, is that you can really knock down cancers quite well, but they come back, and they often come back stronger, which is the other big problem. So you have to understand it holistically enough to get rid of all of it. That's the really hard part, because you got to get down to those last cells. Because if there's a few of these, what are called cancer stem cells, if a few of them are left, they'll regrow your tumor with different genetics. And so it's really, that's what makes the problem incredibly hard. And the other point I'll make is, in the popular press, you'll often hear people will talk about cancer. But I think even the term cancer is a total misnomer. You should basically never use the word cancer. It should always be cancers, in the plural, because cancers are a huge, massively heterogeneous, highly diverse set of diseases and conditions and molecular mutations, and it is not at all a single phenomenon. And even when you say prostate cancer, a lot of people want to move away from even the terms prostate cancer or lung cancer or breast cancer, because what probably matters more are what are the genetic drivers of those cancers. Like your prostate cancer genetically might look more like someone else's breast cancer. And so that, whether it's breast or prostate makes a difference in terms of the tissue that it's around, right? And so there's certain aspects of the environment that will be similar, but it doesn't mean that the tumor itself necessarily has a lot more in common with other tumors from that same site compared to if it had happened in a different site. So as with all these things, it's very complex in the details, but cancers is rightly understood as highly diverse, even individualized. Probably no one has ever had, the exact type of cancer that you have when you have it.
Right. And the cost benefit analysis of treatments. I just recorded a podcast episode yesterday with a woman named Inga Thompson, one of the most decorated American cyclists in history. We were talking about, controversial trans issues of male to female trans in women's sports. It was a women's rights, women's sports conversation.
Yeah.
But she had she's like in her early 60s. I think she's 60 now. Anyway, so she had a breast cancer double mastectomy, and so she's talking about this publicly. And said, so the initial diagnosis was a very small little, like a tumor or whatever in the breast. And so, the doctor's like, okay, here's what we're going to do. We're going to cut it out. Then we're going to do the radiation. We're going to do the chemo. Then every 6 months, you're going to do this, do that. And this get carried out for several years. Hopefully it won't come back. It might come back. We'll have to do this and this and this. She goes, well, what if we just cut the breasts off? Just do a double mastectomy, done with it. And they're like, but the model is to save the breast. He goes, I know, but to do that, I have to do all these other things. So what if we just did the double mastectomy, then I don't have to do any of that, and then I could deal with those issues over here? And that's what she did. And I thought, I never really thought about it like that. That it's sort of a cost benefit analysis. It's astonishing.
Oh, absolutely. And because the pathway, the traditional pathway down, cancer treatment route is pretty awful, right? I mean, people survive it, and I've certainly had family members that have gone through it and survived it, and made a big difference, and that we're very grateful for that. But it is a tough road. It's definitely a tough road, and so people do have to make their own cost benefit analysis of where they're at and what their options are, and it's important to be proactive. So I think that is a good thing.
Well, I hope you guys are right, because at 68 again, I know it's coming. You have a line in your book that, even if you eliminated every single cancer, all cancers, it would only extend the average lifespan by 3 years or something like that, right? So there's just so many other things, including just aging, I guess. How do you think about it?
Yeah, and that's really what that's pointing towards, because I am a big fan of aging research, and I'm glad it's taken a move into the forefront over the last few years, because if we could slow down the fundamentals of aging, and that's a very tall task, but if you can slow down fundamental aging, you could have a bigger effect on lifespan than probably anything else. Because exactly, if you get rid of all cancers, you're still going to die of heart disease. If you get rid of heart disease, they're then going to die of dementias, and there's just kind of a wall, right? When we got rid of infectious diseases, now we all die from cancers, right? There's all these issues that come around, and so I think that aging becomes just really a tantalizing prospect. And so the big debates right now are around what really causes aging, and is it possible to reverse it? So I'll start with a simple example. So at one level, so first let's talk about what we mean by aging, or at least what I mean when I say aging. So aging to me basically means that there's a probability when you're age 20 that you'll die before you're 21. There's a probability when you're age 30 that you'll die before 31 and 70 before 71. And that the odds of you dying as you get older just go up, right? It gets higher and higher and higher. And to me, that the slope of that line is essentially aging. And I think that's the most useful practical definition. I'm influenced by that when I was an undergrad in engineering. I remember they gave us an example that light bulbs don't age. And I thought that was interesting. And what that means is that a light bulb doesn't blow out because the filament gets older. It just blows out as a function of the electrical grid. So a light bulb on day 1 is just as likely to blow out as a light bulb on day 30,000. It just, whatever happens to the electrical grid. So that flatness means it doesn't age. So that's how I think about it in the body too. So at one level, the notion that you can slow down or reverse your aging by that metric is totally non-controversial. Because as you get healthier, so you reduce your hemoglobin A1C, you get out of pre-diabetes towards normal, you reduce your body weight down to a good weight if you're overweight, or you raise it if you're underweight, or all kinds of just basic things we think about will affect your probability of death. So on that standpoint, we can think of aging as just the aggregate of all those health measures. So that's one way in which the notion of slowing or reversing aging is not controversial. Now, the flip side that I want to present, though, is what our dreams are about aging is that, maybe we're 80 years old and we'd rather be 25 and have the body that we had when we were 25 years old and have that in every respect. And that would be like true functional, absolute aging reversal. And we don't have that, obviously. So there's some space in between, like this totally non-controversial, we can make ourselves healthier, our odds of dying go down, the likelihood we'll live longer goes up, and the fantasy of I can be young forever, right? And so people are trying to go down that path to see what's possible. We know that certain compounds and interventions will extend lifespan in animals. Right? These are things like affecting NAD pools makes a difference. Calorie restriction, right, makes a difference. Rapamycin has worked in a number of cases.
Does calorie restriction work in humans? I thought it was mostly mice and didn't replicate so well in humans.
I don't know if we know.
What's the latest on that?
Yeah, my understanding there, I don't know if it hasn't replicated or if it just hasn't been replicated. Because mice we know because they only live 2 years and you restrict them and you go. I am not aware of any long-term calorie restriction study in humans. You can do observational studies, but it's pretty confounded. So I would say we don't know in humans. If you know of someone that has gone farther than that, I'd certainly be super interested in that.
Well, there was that guy at UCLA, Roy Walford. Remember, and he went into the biome 2 experiment and all that stuff. Well, I think he died of prostate cancer, I think it was, or whatever.
But he's an N of 1, so it's too hard to say.
N of 1 also, and also these calorie restriction people, they always look unhappy and miserable. It's like they're always hungry.
Yeah, my favorite
I don't want to live an extra 2 years if I have to spend the previous.
My favorite quip about that, which you may have heard, is the calorie restriction will either extend your life or it will really make it feel like it is.
Right.
You can say it a little better than that, but
how much longer do I have to keep doing this?
Yeah. But you will definitely feel like your life is longer. There's no doubt about that.
But the theory about it is that it reduces inflammation or something like that, because you're just the amount of
less
metabolism.
Less oxidative radicals, less stress on your metabolism. So there is this notion that this is why a lot of people have gotten into fasting. mTOR signaling happens too, and you want that to go on and off. And so some of those cycles have turned out to be important.
You mean intermittent fasting, just every day?
Yeah, intermittent fasting or
12-hour block.
Yeah, so I typically do something like that. I try to eat it within about an 8-hour window. I'm not super strict about it, but I mostly eat lunch and dinner and fast until lunch typically. So there's some evidence that those kind of things can be helpful. Basically, putting your body under stress that you can recover from is good for you. Whether that's cardiovascular exercise, weight training, some degree of fasting, some degree of cold exposure, some degree of basically anything that puts your body into a stressor that it has to respond to that isn't overly much is helpful as you get older.
Oh boy, I love the hot tub and the steam room, but these cold plunges,
Are horrible.
oh
Horrible.
God.
I started doing a few lately just to see because there's so many people doing it, and since I'm in this area, I've started a very mild version of it, I suppose, which is like showering in the morning. I won't warm it up first. I'll just go
Jump
cold.
in. What
It
now? What is it? What's the mechanism it's supposed to do?
it releases these cold shock proteins, and so the idea is that there are certain proteins that will induce repair in our body that you get that release when you get under exposure to cold. And the dosing of this, people are still trying to work out. I've seen estimates anywhere from like 6 to 11 minutes a week or something like this of cold exposure. I have, yeah, the diehards will really do the ice baths,
Yeah. Oh,
and those
brutal.
are brutal, absolutely brutal. So I don't go around recommending those those to everybody. The diehards really, love to do that. Superman and Tia and those guys will all, do the cold plunges and
And have you looked into diets like vegan versus vegetarian versus the paleo all meat? I don't know. I'm all over the place on reading these studies.
I haven't delved terribly deep into the diet space. We've done a little bit of work there. The one thing that I think makes a huge difference, no matter what your diet is protein intake. So the and this is a very simple kind of thing for people to think about, and I've found this to be really helpful. But if you're doing a diet, or I don't even like really the word diet, but your diet, right? Not a crash diet or something. But let's say you're overweight, which is the vast majority of Americans, so that's likely true, and you're trying to lose a bit of weight. Like one of the most important things is as you're doing that in your diet is to make sure you have sufficient protein so that you don't lose muscle mass. So one of the things we
Mm,
know that makes a huge difference in your longevity is how much muscle you have,
mm,
because you're going to go down between 0.5% and 1% per year um, of muscle mass over the age of 30 if you don't fight against that. So that decline, and then when you get older, that makes you more frail. And the huge thing that happens late in life are falls, right? You fall, you fracture a bone, you're laid up, and you're waist away, and that is the signal near the end for most people. Like that's what happens. So balance, strength, these things are massively important. So you want to keep protein level up. And one rule of thumb that I've found to be super helpful is, let's say you're aiming for a certain number of calories. Let's say you're in a calorie deficit and you're trying to eat 1,700 calories. Then, as a rule of thumb, just take 1 0 off and then look to get 170 grams of protein. Now, strictly
mm,
speaking, that's probably a little more than you need, but if you're in a deficit, I think being on the higher side on protein is better. It also keeps you fuller,
mhm.
more satiated, and you get and it also has a higher thermic effect. So if you eat equal calories of protein, it takes more energy for your body to break it down than the same amount of calories of fat or carbohydrates. So
Like
you get
how?
a little
What
benefit
what
there too.
what would that be the equivalent of? Like a steak or a burger patty or 2 eggs or what would be the equivalent?
Yeah, so 170 grams of protein is quite a lot of protein. So I like to start with a breakfast that gives me a really high amount of protein. So I'll typically start with about 8 ounces of Greek yogurt. And Greek yogurt is very high in protein. So if you eat, remember what, so if I eat 300 calories of Greek yogurt, it's like 60 grams of protein. So it's about a
Mm,
2 to 1 ratio. And what I like is that this gives me a sense of, and then the other thing I really like to do is put collagen in my coffee. So I'll put
You
about
do?
20, I really do, yeah. I do that every day. I call it protein coffee. I really like that. So you do, I do it every day. I put collagen, and then I put a little bit of a high protein
What
milk.
What's the product? What would I look for in the supermarket for collagen?
Well, Thorne sells collagen, so there's Thorne collagen, but a lot of
Oh,
places do. So Thorne sells collagen. That's what I use. There's also Vital Proteins will sell collagen, and it doesn't have much of a taste to it. So I like
mm,
the flavorless type. I'll put a scoop in, which is about 16 grams of protein, and then I will put a little bit of Fairlife high protein filtered milk, and I'll put that in, and that gets me a little, maybe a few more grams. So it's about 20 grams of protein. So when I do that, it means that, for breakfast, I basically and when I say breakfast, I usually eat it at like 11 or 12. It's
yeah,
but
yeah, I understand. Me
I
too.
tend to
What
eat
about
two
what's
meals a
that?
day.
Bullet
The
coffee? Is that
bulletproof coffee is a little different. That they'd use grass-fed butter, so that's
right?
that's a keto
Right.
where they go with the fat. Another popular thing to do. I like it because it lets me get to a high level of protein intake. And then if I'm looking at different foods or on packages, I like that one. I try to have an average of one. So I know that if I start my breakfast and I'm up over, and I'm up at about a ratio of two, right? 300 calories, 60 grams of protein. So, 3 to 6, that's the two I'm talking about. So that two ratio, I know that I start from a high place and it gives me a little, so if I eat some fruit or I do whatever that brings it down, I stay higher. And that I've found to be a really useful process. And it's been, because I've been really working on, body recomposition for this, trying to lose fat and put on more muscle, like 95% of everybody. And and I just find that that's, and I got that from a nutritionist, right? That, but I find it to be a very useful
What
rule of
about
thumb.
carbs? Bread or pasta, granola, that kind of stuff.
Complex carbohydrates definitely better than simple. For the most part, I think you want to avoid in the vast majority of your diet anything that has simple carbs in it. So you don't want, yeah, refined sugar, flour. So most breads you don't want, very much of. You don't want, most pastas, and I love all that kind of food. So I'm not saying you like don't eat it, but it
Because
shouldn't be a staple. It's got to be a
those your
minor
body
point.
converts to fat, right?
Yeah. If you're in a calorie excess. Now, if you're in a calorie deficit, one of the things I like about the protein ratio is as long as you make sure you get enough protein, one, you're not going to be nearly as hungry. It makes
Yeah.
a huge difference. But then you can kind of do what you want to with those remaining calories. So you can actually have, pastas or bread. The reason I think you got to be careful with the really processed sugars is because they become neurologically addictive.
Mm.
And I struggled with this for a long time, where you'd feel like pretty immense psychological pressure to satisfy the sugar
Mhm.
craving, knowing it's bad for you, knowing whatever, but it just, at least, I know I feel it that way. I know a lot of people do, where it's just like this gnawing need that's on you
Oh,
all the
I
time.
have
You're
a
addicted to it.
I have like a window, like 8 o'clock to 9 o'clock at night.
Oh,
I'm just like
exactly.
drawn to the freezer to get the ice cream or the chocolate chip cookie and the cut. And it's like if I can make it to 9 or so, then I start to get tired and channel surfing, falling asleep on the couch. I made it, right? It's like, oh, I made it tonight. The craving's gone away. So I have to have some
I'm exactly the
distraction
same.
there.
It's late at night. Late at night is, and I've, but this focus on the higher protein has helped a lot. And I have used some, like special products as well. So ice cream. So like I love ice cream.
Hmm.
Um, and and so far this year, you know, I've I've been doing this high protein, and I'm down about 16 pounds so far this year,
Nice.
and as which has been great. And during that period, I have eaten lots of foods that I like, but in reasonable portions. And I do a number of different things. So one is, like, if I know we're going to go out to an amazing restaurant and I know I'm just going to want to eat, I just fast the rest of the day and I
Mhm.
only eat that meal, and
I
it
do that
saves
too.
up quite a bit of calories. And I find that I can handle that, and I can have a fabulous meal and go out with everyone and just enjoy what I'm doing, and it's fine. And I just time average it, and you're fine. That's a really useful trick. Another thing is, because I like ice cream and I don't, eat it a ton, but I have started having this Halo Top ice cream. I don't have it all the time, but if I really have it, it's not as good. It is not as good as, but since I'm actively working on losing some pounds right now, then it is but it does, but I do like it, and it does kind of satisfy that craving, and it's higher protein, right? It's a higher protein kind of
So
ice cream and
it's
low
not a
calorie.
milk dairy ice cream alternative.
Yeah, it's a little high. I just know it's a little higher protein and lower calorie. It's especially the cookies and cream is pretty creamy. It's pretty good,
Ooh,
but it's not as good as like the best ice cream that
Ben
I
and
like. And
Jerry's.
eventually
Yeah.
I'll get back to that every once in a while because I do really love it. But what I do like is that, so far this year I'm down, 16 pounds or so, and I've had ice cream, you know, a couple times a week or something like that. And
How
so
tall are you? How tall are you?
I'm 5'7".
And you weigh what?
I weigh 179.
Okay, right, because that's my height, 5'7".
Yeah.
Um,
I'm a bit overweight and I'm trying to get it back down.
well, but I don't know, so like your BMI would probably be like 23 or 24, right? But or I don't know,
Yeah,
like
so I'm in
because
the
I'm like
overweight
that.
category. I'm not obese, but I'm overweight. I'd like to be
See, I
down
do wonder
about
about
another
that because
15,
what if
20
you're
pounds.
what I don't know what your upper body looks. What if you're kind of husky and muscular? The BMI is not going to measure that, right?
It doesn't. It doesn't. And in fact, we did a paper for Nature Medicine a couple weeks ago on something called biological BMI. So
Mm,
your BMI is not that accurate. Um, and that's the other thing. Like my weight has come down 16 pounds, but during that period, I have also put on muscle. I've gained 3%.
right,
My muscle
right,
mass has gone up by 3% during the same period.
right.
So, and you have all the devices, right? So you can watch it. And I've been losing very slowly. I'm recovering because the move from academia to industry stressed me out apparently because my body just went up like crazy and I'm just
Mm,
fighting it back down. I don't know what happened, cortisol or something. Um, but it's fighting it back down. But as you go through that, it's biological BMI. So your BMI is so the more muscle that you put on, obviously your BMI goes up, but you're healthier. And so biological
right.
BMI, we did an analysis. This used all the data from the Aravel cohort, and we analyzed all the metabolites and proteins and all these thousands of measurements, and we used it and we tailored it to try to estimate metabolic health along the same trajectory of what BMI is trying to get at. And what we were able to show was that BMI was pretty inaccurate for about 30% of people, and that you could take these measures, and then if you looked at it for health outcomes, cardiovascular disease, um, diabetes, different health outcomes that you would care about, that the metabolic BMI was much more predictive than BMI itself. So this is something that we'll likely deploy, in some form or another, where you can get access to a test that will give you your biological BMI. BMI has the nice element that it's just so dead simple to measure and look at, but it's inappropriate, certainly for athletes or people that put on muscle, and it can falsely let you think you're getting healthier when you're losing muscle, because if you fall in love with the number on the scale, and I've done this before because, in the past I lost, we went through a period and I lost 30, 35 pounds over the course of a few months. I was being very vigilant and trying to do things, but for sure lost muscle mass during that period, which was a pain. This is a problem that people go through. And so that's why I love this notion of you just keep your protein intake high, exercise, lift, do resistance training multiple times per week. And then you can be in a calorie deficit and you can do it in a way that you can gain muscle at the same time.
Mm-hmm. On the
If you've got
protein
excess
front,
fat.
chicken, fish, red meat. What's the research on that latest?
Yeah, I think all those are fine. I think they can be good. I think the best diet is the one you stick to. So eat
Mm.
things that
Yeah, right.
eat things that you like. And that's the other thing I've really been focused on. It's very common advice, but it's good advice because just don't try to get on a diet that you think, I can hold on to this for 3 months. It's awful, but I can do it, because you're going to come off it and it won't make a long-term difference. You've got to find a routine that you enjoy and that you can do. You can do something that you like, and that you'll eat something that you like. So whatever kind of protein that you really love, I'd go for that. I do like doing, I like doing the collagen and, and supplementing some with protein, just because I find it easier to get to as high a level as I want. Red meat, you do have to watch out a little bit for. There are some, like the microbiome test, that I talked about before. So if you have certain bacteria, like it will turn L-carnitine, which is in red meat, and it will turn it into trimethylamine, which your liver turns into TMAO, which is a risk factor for cardiovascular disease. But if you don't have those bacteria, like that won't happen. So one of the things I used to watch when I would eat, if I was eating a fair amount of red meat, that I would, observe whether or not I was getting increased TMAO in my blood, for example, to know if that was, going above a certain threshold. Fish, let's say you're eating, lots of fish or sushi or things like that, especially tuna sushi or something like that. Great for you, really good source of protein, omega-3s, all excellent. But tuna is pretty high up the food chain. It is a repository for mercury. So when we were doing our studies, we had multiple people who really liked to eat that, and they were getting unhealthily high towards mercury poisoning. And so they had to back off that. So there are different things that you can do subtly. So you probably want to vary up your diet. Is if you're not testing, that's particularly important. But
Salmon
those
would be
can all
better
be
than
good
tuna.
sources. Salmon would be better than tuna. It's not quite as high up. Yeah.
And the latest on exercise, I presume cardiovascular. You mentioned weights. The weights because of stressing the bones and joints to strengthen them.
Yes, you definitely want to strengthen the joints and the bones and the ligaments, and putting on muscle mass, because like I was mentioning, you lose between 0 and 1% of your muscle mass per year. So decade over decade, that really starts adding up. So you become much more frail in your later years. And so that power of having muscle, and you want it to be functional. So some of the things you really want to be able to do is you've got to be able to stand up from the ground without any assistance. That's really important. And when you're young, that's easy. And when you're older, it gets harder. And if you lose that, it's really bad, because then you're putting yourself at much more risk. Balance postures. So things like yoga, balance postures, to go along with strength training, super important. Stretching, the range of mobility becomes really important, because if you don't move your body a lot, and you're not putting it into spaces, then you lose that ability, and you'll become more and more rigid and more fragile. And so, yeah, my wife and I just started this program called The Science of Stretching, which is a little bit of a more aggressive type of this, but it's 15 minutes of stretching a night, which sounds like very relaxed and easy, which is kind of what I was expecting. And but what it is it's 3 poses, and you hold each of them for 5 minutes. And
That's a
by
long time.
minute 5, it feels like
Yeah.
a lot.
Yeah.
But it makes you flexible. Within a few weeks, it makes a pretty big difference. And but there's a lot of things you can do. So, and yoga, even restorative yoga, things like that, just to have range of motion, all those things make a difference. And then for dementia, it's really important that you keep oxygenation into your brain at a high level. So that's cardiovascular fitness. Elevated VO2 max is super important.
Yeah.
Elevated VO2 max is really important. And one of the exercises that came up in a paper that we've been going through that was quite interesting also suggested that exercise that where you jump up and down, one, because it really gets your heart going, and you're doing well that way, but that was the strongest associated exercise with reduction in dementia. And so maybe it's something about just going up and down that pumping your blood up high or something. I loved that, because a few weeks before we read that, I had put a trampoline in my living room, and so so if I'm watching a game, especially if I'm watching sports, I find that if your adrenaline's kind of going, you're watching the fourth quarter or something. I like to bounce during that, so it's a good way to just kind of work exercise
Or
into your
maybe one
daily
of those
life,
big yoga balls, the air ball, and you just
or the
bounce on
big
it. You sit,
or the
sit and
sit
bounce.
and
Yeah,
bounce on it. You can sit and bounce on
interesting.
it. Yeah, all those
Right,
kind of things.
right. All right. So speaking of dementia and Alzheimer's, what can we do to prevent this? I was just reading about that. There was a recent story about, the plaques and tangles. Okay, plaques and tangles. That's the problem. Well, so there's people that have had autopsies that had Alzheimer's and had no plaques and tangles. Others that had autopsies, plaques and tangles, never exhibited Alzheimer's symptoms. So obviously there's other factors and probably many. What's the
Yes.
latest on this?
Yeah. So I have a pretty strong view on Alzheimer's disease, and so I want to just explain that a little bit, and then I'll get into what I think you can do for prevention. So to start with, I think that we have really been on the wrong track in Alzheimer's for a long time. And I talk about this, or we wrote this up into the book,
Hmm.
and it's one of the things we've been developing at Thorne over the last couple of years is a complex systems model for how the brain maintains health. And so this is building feedback loops about how the brain maintains energy, how does it clean out debris, all these different processes into a complex model. And then we've used that to simulate 10 million digital twins. And a digital twin is a representation of an individual person's biology. And I'll come back to that a little bit in a moment. But building this thing has totally changed my view of what I think is going on in Alzheimer's disease. And and we're able to quantitatively predict a huge amount of data out of the literature. So it uses data as we build up the molecular pathways and so forth. We use data from insights from about 950 papers. And then we do simulations against populations and we compare that to over 30 human clinical trials and research studies. And so this is what has emerged from that that's different. So first, what people almost always say is that Alzheimer's disease is caused by amyloid plaques. And I don't think that's true. So, and we'll get into that. So what I think is the biggest factor actually hinges around metabolism and what the brain has to do to keep up energy. So your brain uses 20% of your body's energy, and it's 2% of your body's biomass. So it's 10 times more metabolically active than the average of your body. So every second of every day, you got to feed energy up to this thing all the time. And so one of the problems that happens is that as you get older, your ability to perfuse oxygen in your brain through your blood vessels goes down. And this has all been measured. So it decreases, just like muscle mass, right? It's getting worse. And as that happens, there are certain regions of the brain that will become lower in oxygen. And when we run you run the calculations in metabolism, this is what my PhD work was really about. But as you run these calculations on metabolism, there's an inversion where the amount of energy that you can create goes below the amount that you need, and certain neurons will start dying. And then as they die, you put more demand onto the remaining neurons. So their demand goes up and their supply is staying there. And so as they go up, then they die, which puts more pressure on the rest, and you get this positive cascade of cell death. Okay, so that's the simple concept at the heart. Now, the second big factor that everyone knows about Alzheimer's is that there's a gene, ApoE, and if you have ApoE4, you get high risk of Alzheimer's. 3 is normal, 2 is protective. So one of the things that happens is that when we run these simulations and you identify, how do you keep neurons making a lot of energy under these low oxygen conditions, it turns out to be really important what's happening in the supporting cells, which are called astrocytes. So you have about 9, I think it's 9-to-1 ratio astrocytes to neurons, and they support energy generation in the neurons. So when we run this, it turns out you want to keep cholesterol level in the astrocyte low in order to keep energy generation high in neurons under low oxygen conditions. So ApoE. So ApoE has a role in the transport of cholesterol out of astrocytes. ApoE4 does it slowly. ApoE2 does it fast. And if all you do is take those two facts, the lowering of oxygen and the difference in the efficiency of keeping positive energy balance under that condition, you can recapitulate the ages at which all the different genotypes get Alzheimer's disease across the country. And you can get it within a very close accuracy. And that's against a whole range of genetic and environmental backgrounds. So that's the next layer. By the way, I could go on to this like 14 layers, but I don't want to get us overly bogged down. But that gets at the heart of it. Then you can dive into other genes that are behind Alzheimer's. There's a gene called TREM2, and it has to do with the energetics of what's required to clear debris. So as these cells are dying, you get all this debris that you've got to clean out. Well, that comes at an energetic cost. So it's just like you're running a business, right? And you have cash inflow and cash outflow, and you're just counting it up. Every one of these things costs energy. And if you spend energy there, you don't have it left to protect it. And so we just do an accounting like this. And one of the things that happens then is as your neurons die and you lose synapses. So as you lose that synapse firing, as it goes down, you cross a threshold where you can no longer do what's called Hebbian learning. This is what our brains do, right? What fires together, wires together. But you don't have enough firing to learn on top of. So the brain has to secrete a molecule in order to enhance, to recruit additional synapses so it can do Hebbian learning. And what is that molecule? Amyloid beta. So it recruits, so amyloid beta is brought in by the brain or made by the brain in order to recruit these synapses so you can keep cognition. Now, as a byproduct, these things are going to glob together and you're going to get those amyloid plaques. The plaques aren't the cause of the disease. Now, it gets complicated because they do cause some problems. Like amyloid can embed in your blood vessels and it will constrict your blood vessels, which comes back to the central problem. It limits your energy because it's limiting your oxygenation into the brain. And so it can look causal sometimes under certain conditions. But anyway, we put that all together into these 10 million digital twin simulations, and we're able to recapitulate a huge amount of diversity of the effects of statins on the brain, the effects of low vitamin D, bad sleep, high inflammation, all of these different elements in one unified mechanism that we quantitatively simulate. And we're going to release this as a product, at least for supplements and lifestyle recommendations. And we'll look at it through regulatory to get it out for drugs as well. But it's an ability that I think is pretty remarkable, and it gives a different view of what's going on. And then I'll get into prevention, but I'll pause there since I've been going for a while.
Well, so you mentioned statins in that list of bad things, right?
Actually, no. It's a little more complicated than that.
Oh, well, because you said something like not enough sleep and so on. The statins.
So statin. Yeah.
No, go ahead.
Yeah. So the statins is actually a really interesting case. So there was a trial that came out. This was about a year ago. So we had built the beginnings of this model and the view that we had of Alzheimer's at that point. And then a new clinical trial drops. And unbeknownst to us, until it came out. But so we read the paper and it says lipophilic statins. Lipophilic means fat loving. So these are the kind of statins that can get in your brain because they can cross the blood-brain barrier. So it says these lipophilic statins accelerate the rate of decline in Alzheimer's disease. That's the headline. It goes everywhere. Right, big deal. Now that study was fascinating to us because they had done 8 years of PET scan imaging. And what they found was that they reported that they found these areas of hypometabolism, which means low metabolism, in certain regions of the brain. They said these are the areas that died first, which is exactly what we had predicted under our model. So this was just very clear evidence, pictures of it, right? Amazing. And they centered on the same mechanism that we had, which was this cholesterol in the astrocytes, which are being affected by statins. And so we thought that was fascinating, except for one thing, which is that statin should keep cholesterol levels in the astrocytes lower. So we thought they should be beneficial and not harmful initially. So we were a little perplexed by that. But this is why you don't do research by headline. You do it by going into the data, right? So we go into the data, and what we find, and we figure out, okay, who exactly is being recruited? Because these models let us simulate cognition over the course of your entire adult lifespan. So we can look at different areas in the curve. So what we saw was that, well, just to back up slightly, there are other studies, though, that said people on lipophilic statins got Alzheimer's at lower rates. They got Alzheimer's less than people not on statins. So we had papers that said statins good, and we had papers that said statins bad. And they were all looked to be well-run, good studies, right? We talked about reproducibility. What's going on? So because the model is amazing this way when people understand it, because you have this lifetime cognition that you're able to simulate. So we can simulate what's happening in your 40s, your 50s, your 60s, your 70s, your 80s, your 90s. We can all of those things. And so what happened is that when we delved in, we saw that in the model, when we put in the mechanism of statins, it changed the shape of the curve. So what it meant is that if you were going to have kind of gradual decline in dementia, if you were on the lipophilic statin, it changed the curve so that you stayed higher for longer. So it delayed the onset of what's called mild cognitive impairment. But if you went from mild cognitive impairment to Alzheimer's, it dropped faster. So it changes the shape of the curve. It stays up higher and then it falls faster.
Right.
So it's like the blind man and the elephant, right? You come in. So
Right.
if you do the study early, you say
Measuring.
they're good. You do it
Right.
later, it's bad. So on that standpoint, but I think on the whole, they're probably good for dementia, because I would take the delay in mild cognitive impairment. And once you recruit into your study people at mild cognitive impairment, if they were on the statin, they're probably later stage in the disease. And so I don't know if it's a real thing that the statins are accelerating their decline, or if it only looks that way because it was propping them up and now it can't work anymore. So they're at the end and they fall off a cliff. But that's how this thing is just so useful to us in just contextualizing this kind of information.
Yeah, this is amazing. This is really interesting and important because, yeah, the old model doesn't seem to be making the kinds of predictions we'd like to be able to treat it in the example I just gave of the plaques and tangles. Okay, so if what you're saying, if I'm understanding this right,
Mm-hmm.
so cardiovascular exercise, anything that gets more blood with more oxygen pumping into more of the brain, that's going to delay the cell death.
Yes.
And so, yeah, because there are studies, right, that show cardiovascular disease is good for not prevention, but just slowing the decline, I guess, of all kinds of dementias.
Well, and there was a big study called the finger study out in Finland that showed this, right? That you could delay the onset of dementia through lifestyle, diet, supplements, things of this nature. The only really successful trial, I would argue, in Alzheimer's disease. There have been a couple of drug trials that showed some modest effects in decline over the last few months. But if you look at this, so here's things that people can do. So exercise is important. And under the model that I just described, it's obvious why that's true. If you're really focused on amyloid, I mean, exercise is almost always good, but it's kind of mysterious what it does to amyloid. But it's very clear what it does when you think about this from an energetics perspective. You just keep your blood flow up. The second thing is that we find that when we run through the digital twins, what happens when you are in this hypoxic condition, you're trying to maintain energy, is that phosphatidylcholine becomes rate limiting. You run out of it. And so the hypothesis there is that, and choline is kind of well known as a factor in dementia prevention. But so we look at this for phosphatidylcholine, and so we had this hypothesis. So then we go into the nutrition literature, and it's always dangerous to just straight up machine learn the nutrition literature. But we go in there with one hypothesis, and we just ask the question, do people who eat diets rich in phosphatidylcholine get Alzheimer's later than people who don't? And if you go into those papers, the answer is yeah, on average 3 years later. So there's an estimate of an effect size. That's what we calibrate to. So we think that, and we think with supplements and so forth, then you could push, eggs is like the primary dietary way that you would get phosphatidylcholine, and you can push that higher if you'd like.
Oh, nice.
And so and so great, right? And so you get that, and so that has an effect. Low vitamin D leads to earlier dementia, so you want to, so watch for that. High inflammation, right? So you want to keep your inflammation lower, so control for that.
Okay, hang on.
And so there's a whole bunch of features like that.
Vitamin D can get supplements, or you just spend more time out in the sunshine.
Yep, time out in the sunshine, or take a pill, right? They're cheap and
Yeah.
easy, but either way.
Right. And eggs and protein. Okay, that's good. And then what was the other one? You just said something else after the
What are some of the other ones?
vitamin D and what was
Vitamin.
the one after?
So measuring CRP or something like that with inflammation. So if you have
inflammation. Right. So how
high
do
inflammation.
Yeah. Right. How do I do
So
that?
so there are different. You can take different products or different foods that are anti-inflammatory, and so just looking those up and getting them. NAD pools looks like it may actually play a role as well. There was a recent paper that just came out on that. We're starting to look into that, but for that you can take nicotinamide riboside or things like that will have an effect on your NAD pool. What was the other one I was going to say? Stress. Stress is another big one. So measured like cortisol levels if those are high. So anything you can do, and again, 3 options: meditation, read a book, sociality. It's a harder one to
Right.
model, but the biggest predictor of longevity is time with friends and
Right.
family, and sociality, and having
Right.
a good
I
reason to
love
be
those
alive.
studies.
You know,
Yeah.
that's what
Yeah.
matters.
I had the guy Mark Schultz on the show, his a book on the good life, that Harvard longitudinal study since 1938 they started. Right. So
Yes, yes.
better than not smoking, better than exercise. You don't have to eat kale and run marathons. You just have to have friends.
It's
That's
the number
easy.
one thing, right? It's the number
Yeah.
one thing.
It's astonishing. Yeah, and the stress thing,
Amazing.
probably social media is not good for stress, particularly for teens. We're staying on top of this debate about, well, how come there's this massive spike in depression, anxiety, cutting suicidal ideation and suicide amongst teenagers just in the last like 10 years, even less than that. And, so why? Social media is one of the culprits that has been fingered, and screen time, and because that leads to less time with other people, less time outdoors, less vitamin D, less exercise, right? So all those things.
All those things.
Yeah.
And it makes a big difference.
What about brain games? Do I have to do Sudoku puzzles? I don't really like those. How about chess? Can I play chess? Can I just read books and write books and play chess and do cognitive stuff like that? What works? What doesn't? What do we know?
Yeah, so there's no doubt that there's benefit to being able to do brain training of various kinds, or just keeping your mind active, right? Everything that you do, having sociality matters a lot, all those kind of things. There is one specific form of brain training that I think has special merit, though. So the person that I'm thinking of primarily, this was all developed by Mike Merzenich at UCSF. And he's a winner of the Kavli Prize, basically the Nobel Prize for neuroscience. He's really an amazing person who has pushed the avenue of neuroplasticity maybe farther than anybody. And so what I think is impressive about what he's done and how he's turned this into something that's useful for people is that he has created a company called Posit Science, and they have a product called Brain HQ. And what differentiates this from all the other kind of things that we do in our daily life is it deals seriously with the notion that your brain is a physical object and it has certain limitations to it, and that you can improve those by exercising it with progressive overload, much like you would do with a muscle. And so what Brain HQ does is it forces you to find out what is the absolute limit of how quickly you can detect an image in what fraction of a millisecond. How far is the peripheral vision that you have? What's the limit of your short-term memory, and so forth. And so what it does is that it forces you to go right to that limit, and then it does a little bit of progressive overload to try to push you out as far as you can go. And then you take a break, right, and you come back to it a few days later, and it'll push you to try to get you to be a little bit faster a little bit
Like
quicker.
weight training.
It's like weight training for your brain. And I think it's pretty unique that way because most things we do, they're taxing, right? When sitting down and writing the book was a lot of mental effort, and our jobs are a lot of mental effort. But I don't often push myself to the absolute limit of my visual field or the absolute limit of my memory or the absolute limit of my detection ability and so forth. And so that I think is really fascinating. And Mike has done like 200 papers now on the effects of this, and really puts forward a strong case that you can, if you reduce the age of your brain essentially, but get back to a level that your brain fires much more like it did when it was younger. So the brain is plastic that way, and I think those are super helpful.
So the brain is plastic if the neurons are still alive. As you mentioned, if they're dead, you can't bring them back. Is there like a gray state in between where they're sort of dormant, not getting quite enough oxygen, but they're not dead, and that you can jumpstart those again through, again, exercise or diet or these brain games?
Yeah, for sure. Because there's a lot that goes into your neural complexity above and beyond like just whether or not your neurons stay alive and the whole story around dementia that we just talked about. And so for most people, your raw cognitive abilities peak, I think, right around the age of 30. So, and then exactly, not good news for either of us here. Now you make up for it because you know more things, your knowledge is better, you chunk much better, right? You don't have to think through from first principles all kinds of things because you've been through it before. Now that does make us more rigid in our thinking because we have chunks that we already believe in, and so it's a superpower and it's a liability, right? It's two sides of that coin. And so the idea with the training though is that you can capture some of that. And so if you can keep your brain plasticity and your raw cognitive power as close as possible to what you had when you were younger, that's a superpower when you're older because you also know so much more. So if you can harness experience plus more raw firepower, that is an incredible advantage.
And so how do you distinguish between Alzheimer's and, say, more general dementia or senility, or what do those words mean and how do they differ?
Yeah, so Alzheimer's is a form of dementia. So dementia is a broader term. Alzheimer's represents, by far the largest type of dementia. And so it is the biggest chunk. I find those terms, like dementia, I feel like is the term that matters more in a sense because it is talking about loss of cognitive power. Alzheimer's, like I talked about, it's largely driven in diagnosis by amyloid plaques. And so the secretion of amyloid is largely what defines Alzheimer's. There's other things around it, but pretty much they won't take a gold standard Alzheimer's diagnosis until you see the plaques. And in fact, often they won't do a gold standard version until autopsy because there's a lot of controversy about exactly what makes it up that way. But because I don't view amyloid, I view amyloid as a bit of a misnomer in terms of it being taken as the cause of Alzheimer's. I don't actually love that as a differentiator that well. So I feel like dementia is what you actually care about, which is the loss of cognitive ability. And as a person, I don't know how much you care what is driving that, other than if it helps you get better, right? If it helps you stave it off, then you care a lot. And so that gets to be interesting because we do have people that have their heads full of amyloid and they're cognitively fine. In fact, we used to study these a lot when I was working with the NIH and the Mayo Clinic, our close collaborators at the Mayo Clinic and the University of Florida, Nilofer Urtek-Tainer and Todd Goldie. And we were studying what was called pathological aging. And that was a fancy term for head full of amyloid, but no cognitive issues. So there's a lot of people that are diving in and studying this, and it gets pretty arcane pretty fast in terms of like what all the different cutoffs are for different people. But at a high level, Alzheimer's is just a subset of dementias. Yeah.
Yeah. Well, I hope you guys are right about the speed with which these things are coming online. You have a few more decades than me.
We got to get this done ASAP. Fast.
But the baby boomers, I mean, that's a massive population, and they're going to be hitting the wall here. And so this Alzheimer's thing is going to be huge. I would imagine anybody could figure this out. That's a couple of Nobel Prizes and a couple of Kavli Prizes there.
If you can get it implemented in a way that really makes a difference in people's lives, it's very important.
Yeah, for sure.
And it is the big problem that's coming, right? With the aging of the population, the progress we've made on other diseases, there's no point in extending your lifespan out a long way if your brain isn't coming along with you. So it's absolutely critical that we solve this.
I read quite a bit of the literature on longevity attempts at life extension for my book, Heavens on Earth. And, I met Aubrey de Grey, the life extension guy. The first person to live 1,000 years is alive today. It's like, yeah, okay, where is he?
I volunteer.
Yeah, I mean, he's quite the character. He's got that SEN system, the 7 different things that you can solve inside the cell and all that stuff. And I don't know if he has an elixir to life. It apparently is beer. He drinks a lot of beer. I hope beer and wine is okay. By the way, what is the research on that? I mean, I have like a glass of wine or two with dinner, or maybe a beer in the afternoon after a long bike ride or something like that. I don't abuse it, but what's the research on alcohol?
Yeah, the very unpopular paper that came out recently was a huge meta-analysis on alcohol and basically concluded that all amounts of alcohol are detrimental to health. Everyone wants to believe that it goes that it's beneficial and then only at the end.
Yeah.
Now that said, small amounts of alcohol are not hugely detrimental, and to what we talked about earlier, if they help your sociality.
Oh, right. Stress.
And stress is a negative, and lack of social interaction is the number one predictor. Number one predictor of longevity is social interaction. So So if those positives are coming with it, you could look at that. But from a pure health perspective, I'm afraid that the most conclusive evidence so far that nobody likes is that it's
don't drink alone. That's the bottom line.
Don't drink alone. Yes, exactly. Make sure you get your sociality benefit at least out of it.
That's really funny. Yeah. But It does seem like even you add everything up, it's an asymptotic curve. At some point, you just hit the upper ceiling. I don't think anybody's made it past 120 ever. And even those are debatable. Maybe 115 is probably the upper ceiling. I mean, my sense from reading is that you'd have to change, not 1 or 2 or 6 or 7, but like 1,000 things or something to keep going, to make it to 150, 200 or whatever. And even there, my sense is that we have no idea. If you got to 150 years old, there may be things we don't even know about that would all of a sudden come online to cause problems.
Yes. So, as we talked about aging, trying to flatten that curve and make it so that we're less likely to die and extend that is a huge, that you can absolutely do to some degree. But you're exactly right. There's this barrier that we don't know if we can cross it. We know we can increase health span. That's why we mostly talk about the enhancement of health span, because that's for sure doable. The period of time in which you live a healthy life.
Yeah.
Lifespan, as you hit that end point, is a big dream, of course. And this is where you get into areas that are controversial. Now, the most famous person in aging by far as an aging researcher now is David Sinclair. And the reason for that is that he's put forward this, epigenetic theory of aging on the notion that you could rewrite epigenetics, and that by doing so, you could reset your cells to a younger state, and that all these other issues will follow from just doing that epigenetic reset. A lot of other people take a different view, which is that there are myriad causes of aging, some of which, for example, if you just look at at enzymes that are catalyzing metabolic reactions, as you get older, the number of metabolites, these small molecules that are formed, that are not like the canonical one that it's trying to make, because it will make the one, but then there's different offshoots that you'll get in small quantities. Those get larger and larger as you get older. And there's many processes like that, right? That it just becomes more diffuse, more regulated. and it goes on. Now, one of the arguments, just comes from, decay or entropy. We are open systems, so there's not a second law of thermodynamic imperative that we do that. So we, if we could do control, we should be able to, to overcome that ultimately. But it requires energy, it requires information. And there are these really fundamental concepts in physics around conservation of energy. And information is actually quite interesting. Like, one of the most interesting facts I find in all of science, you're probably familiar with this, was the Maxwell's demon. You remember?
Yeah.
Where you could take something cold and use it to heat up something hot, because you have a little demon that monitors the gas particle, and it opens it the second it comes across, and then you close it, right? Which is so fascinating. But what I found stunning, absolutely stunning, is that when you do those calculations, it turns out that the amount of information that you have to put in the demon is equivalent to the amount of entropy that you are overcoming by doing that. In other words, they had derived two concepts that they called entropy, right? Information entropy and entropy, from an energetic point of view, thermodynamic entropy, and it turned out those things were equivalent. So this is, getting a little more esoteric, but as we try to keep our bodies alive, it's really about preserving this core of energy and information. And, there's fundamental processes that have to happen with that. One of the great biologists I used to work with was Carl Woese. And he's probably not a familiar name to most of your listeners, but Carl's the guy that discovered the archaea, who came up with the idea that there are three branches of life. He won the National Medal of Science for that. I have some stories about him, because he had the office across from me in Illinois, and he passed away a few years ago. Just a wonderful, wonderful, deep man. But he had a metaphor for life that I really loved, which was, he said that the best metaphor for life is a whirlpool, and he loved this notion, because a whirlpool has a structure to it, right? But the water flows in. You can see the whirlpool, you can see its shape, it changes a little bit, and the water flows out. But every molecule in your body is different than it was 7 years ago, right? You have molecules flowing in and out all the time. So what is you? What is the whirlpool? And you are essentially, a swirl of coherent information in this pool. And so it gets down to, what does it take to keep that going and alive and functioning when you start having decay away from that central information? So it comes back to, can your body remember or get back to that state, both from the standpoint of the information it takes, and then the implementation in the physical world of the energy and mass that it takes to make that happen.
That's a great analogy. That is right. The whirlpool of information that is you, not just your genome, but your connectome and all that. Although here I had to, I think I debunked the people that want to achieve immortality by uploading your connectome to the cloud. Or if you want the whole body thing, you'd have to, replicate every cell in your body and so on. And a clone and all this stuff. But the problem is, it's not you. It's just a copy of you. Because let's say you slid the body, your brain, into a scanner and scanned the connect. We can't do this, but let's say you could.
Right.
Uploaded the file into the cloud, and then slid you back out, and they go, well, congratulations, you're now up in the cloud. And you'd be like, no, I'm right here. I'm looking through my eyes. I'm talking to you. I'm here. That's just a copy of me, right? And so we just have to redefine you as multiple yous. Well, yeah, but it's the point of view self that's the one that counts, not just your memories, the memory self, because the memories are always changing. I'm not the same person I was yesterday or 10 years ago or 50 years ago or whatever. Those memories have changed and been edited and so on. There's just kind of this fuzzy set that represents me. But from the point of view self, I'm me right now, and I'm this next second, the next moment. And that continuity has to continue, so it may be cryonics, where there's a break, like going to sleep or under general anesthesia, but it's 1,000 years or whatever, then they wake you up. It would only work if you actually had the continuity. You woke up just like from general anesthesia, you're a little groggy, and then the memories come back online and you're off again. Anyway, I find this a whole fascinating thing, because everybody loves this subject because they want to keep going. right?
Yeah, because you want to see the next that you're invested, right? I want to see what happens in 2100 and 2200 and 23.
right.
I want to see it.
So when these people would say, "Don't you want to live to be 500, Shermer?" I just get me to 90 without Alzheimer's, 100 without dementia, my body falling apart. Because you asked that in the book, would you like to live to be 120? Most people go no, because they're picturing themselves in a bed all wired up in the can. Of course, no one wants that. But if you could be like you are right now for the next 100 years, yeah, okay, I could find things to do.
Yeah, it's always about health span. Because I run into people all the time that say, "Oh, I don't want to live longer." And initially, I found that really,
Right.
you're okay, but it is because of that end of life, and that's a whole other topic, of course. Because the way our healthcare system is set up, we spend a huge amount of our dollars on extending the absolute worst part of life, which is that end part. And yeah, that gets into whole other kinds of issues. But that situation is, if you Atul Gawande wrote the best book on this, the Being Mortal book.
Oh yeah.
Being Mortal, which is such a great title.
Yeah.
An amazing title. And you look at that, all of us say that we would rather die at home with our families, a little bit shorter, but not going through that. And yet 90% of us will die in a hospital with our life, kind of dragged out, in pain. And that gets into a lot of societal issues, because we basically force people into that scenario. At least, our society is set up for that.
Yeah.
And it's really a and I think about that person, my paternal grandfather. I remember, he had a rough last few years going. He basically couldn't speak for the last 3 years as he's in the hospital, and he was only able to eke it all the. When I visited him, he was only able to eke out 4 words that I heard over those 3 years, and they were, "I want to die." That was it.
Wow.
That was it. And so I look at that end of life, and I just think I'm actually maybe I'm on the more extreme end of this. Or more rational, depending on your point of view. But I think, the most part, I believe in a right to die and a right to try to pass if you're in that kind of situation. I think that it's,
as long as it's not abused. Yeah.
As long as it's not abused, you got to have, I'm more for the right to live, but I do think,
By the way, my rebuttal to people like your friend is, I don't want to live to. Okay, then how about we end it tomorrow? Well, no, not tomorrow. No, I got things.
No one wants to die tomorrow. Yeah.
Yeah. How about next month? Well, no, I have things for the next year. Maybe. Okay. How about next year? Well, no. Okay. One more decade would be fine. Thank you. It's like, okay.
As long as you're enjoying the ride, let's try to keep it going. Yeah. Yeah.
All right, Nathan, that's a good place to end. We went almost 3 hours. That was epic. Just so much good stuff in there that I think people really, they want to hear, because this is what we all are concerned about. It's huge. So let's hope your programs all come online soon.